dissolution and diffusion of the nutrients present within the matrix of bead. And the diffusion barrier is being created by the binder that we have used to prepare the beads. So the pot experiment shows good biomass and root proliferation with both the fertilizers, and recommended dose is also essential along with that. And the slow release mechanism is already explained in previous slide, where the zone of unsaturation is the driving force for the nutrients to release. And after a certain point, the release will stop even if the bulk amount of water is present. So it will help in reducing the losses of nutrient that is happening conventionally in the conventional fertilizer, commercial fertilizer, because they dissolve really quickly in the presence of good amount of water and responsible for the runoff losses. And of course, we are placing these nutrients close to the root zone of the plant, so it is increasing the nutrient use efficiency of the plant. So the further progress is that we have already signed an agreement with the company called LCV Fertilizers for further development and commercializing the product. And we are also in conversation with an Israel-based company, that is ICL. So we are looking forward to work more and develop this technology already. So in the end, I would like to thank everyone I have listed in the slide, and many more people are to be thanked. But of course, this place is not sufficient to say that. And at last, AgriVision committee members are like, I'm highly thankful to them for inviting me over here and letting me give this talk. So thank you so much.
Hello. Thank you so much, Avilasha ji. And it was really lovely. We know many things about biochar, its characters, and yes, the two fertilizers that has been slow release fertilizers, MAB and SAB. And thank you so much. Next, we have one more person with us, and many people has been connected with us through Zoom. Thank you so much, everyone. You are on Zoom meeting, and those who are in Zoom, and we will be doing the session, virtual session shortly. Once we complete, once again, welcome you all. And we are here in associated with Society and for Agricultural Research and Management, and in collaboration with Centurion University of Technology and Management. So next, I would like to request P. Sushri Shamli on a topic describing the regulation of nutritive value and abiotic stress response in Moringa oleifera through genic approach. P. Sushri Shamli from Institute of Life Science, Bhubaneswar, India. Everybody, please put your hands together for Sushri.
working with Professor Pulak Kumar Mukherjee, and today I'm here to present a part of my work entitled Deciphering the Regulation of Nutritive Value and Drought Stress Response in Moringa oleifera. Before starting, I would like to thank the organizing committee of AgriVision for giving me the opportunity to present here. Okay, so before starting off, I would like to give a brief of what our group does at ILS. So we are basically focused on generating genomic resources for underutilized crop species, bioprospecting for novel genetic combination for abiotic stress, and developing transgenic crop systems with the aim to contribute to sustainable agriculture, food, and nutrition security. So now jumping straight to the problem, all these newspaper clippings, they show how climate change is now the biggest threat to global agriculture and food security. So the figure here, it shows the vicious circle of the environmental outcomes of climate change. So as you can see, when there's increase in carbon dioxide, the increased temperatures, increased in desertification, increased droughts, the rising sea level, and that causes increased floods, salinization, erratic rainfalls, and this increased intensity of storm. And all this has direct impact on agriculture. So there's increased disease severity in the livestock and crop. There's lower crop yield and lower nutritive quality. So when we are talking about environmental changes such as cold, heat, flooding, drought, salinity, heavy metals, they all come under abiotic stresses. Now, abiotic stresses, they lead to great crop yield penalty worldwide. And out of these stresses, heat and drought, they are major abiotic stresses that severely affects crop productivity and distribution. According to a report by UNCCD, up to 45% of the world's agricultural land is subjected to frequent drought. So, with the problem in hand, what could be a possible solution? A part of it could be solved by the use of knowledge of genomics to generate climate resilient crops with improved nutritive quality. So, whenever we talk about functional genomics, the first thing that comes to our mind is the use of 5Gs of genomics that are vital to accelerate crop improvement. The first G is genome, where a genome assembly should be available for a given crop species to start any kind of research work. Then second G is germplasm that needs to be characterized at genome and agronomic level. Third G is genes that needs to be identified and functionally characterized. Fourth G is genomic breeding that can be used using genomic knowledge and approaches. And finally, the fifth G, that is gene editing, that can be used for improving plant performance. Now, the genetic resources, they can be used to develop the genomic resources, either through genome sequencing and transcriptome assembly. Both the data from genome assembly and transcriptome assembly can be used for gene expression analysis, and from that we can identify our candidate gene. And once we identify our candidate gene, we can either go for gene cloning or marker development. And if we are going for gene cloning, the next steps are functional analysis and transgenics. And if we are going for marker development, the next steps are identification of linked markers for the target trait and then marker assisted selection. So whatever path we choose, the end goal is to achieve improved or new cultivar that is field tested. So our lab, we are mostly focused on the gene cloning part. Now coming to why we have chose moringa for our study. As you all know, moringa is commonly known as drumstick tree, which belongs to the family Moringaceae. It is a versatile plant and is a repository of essential phytochemicals present in every part of the plant. So it's a known fact that it is deemed as a superfood because of its highly nutritious profile and medicinal properties. However, what's a lesser known fact that it can grow in low water conditions and is relatively drought tolerant. So the first thing that we needed to start our work was a comprehensive reference grade whole genome sequence of moringa. And we were successful in decoding its genes. So to prepare these samples, the seeds were collected of Moringa, a Bhagya variety. They were obtained from Krishi Vigyan Kendra, Dhenkanal. And these seeds were then grown in soil in controlled greenhouse conditions. Then the young leaves were collected and surface sterilized and stored at -80 after freezing in liquid nitrogen. Then high molecular weight genomic DNA was isolated, and we went ahead with sequencing using PacBio SMRT and Illumina HiSeq platforms. The long and the short reads were then assembled with Mazurka. Now, whenever we do whole genome sequencing analysis, these are the 4 components that are associated with it. The first is quality assessment, where we estimate the completeness and redundancy of genomic data by providing biologically meaningful metrics that is based on expected gene content. Second is functional annotation, which describes our knowledge of the biological domain. The third is orthologous group analysis, where we delineate genealogy of the genes to investigate forces and mechanisms of evolutionary process. And finally, simple sequence repeat identification that is used to generate integrated maps for plant species and to provide breeders and geneticists with a tool to link phenotypic and genotypic variation. Now coming to the assembly statistics. So from the PacBio sequencing platform, we generated 23 GB of data, and from Illumina sequencing platform, we generated 90 GB of data. We achieved around more than 300 X coverage, which is around 90% of the Moringa genome, since its genome size is 315 MB. So the clean reads were then assembled into 915 contigs with N50 value of 4.7 MB. And further, to check the genome completeness, we did BUSCO analysis, where 95.9% were complete BUSCOs and 94.4% were complete and single copy BUSCOs. Usually a score of more than 95 is considered a good assembly. So next we identified the genes in Moringa genome, and we predicted 32,000 protein coding genes, which came down to 23,757 after UniProt annotation. Now coming to the functional annotation, we assigned the gene ontology terms to the predicted protein coding genes of Moringa using UniProt SwissProt database. And figure B, here it shows the Venn diagram that depicts the annotation of 21,634 protein coding genes of Moringa oleifera with various databases such as PFAM, UniProt, COG, and KEGG. Now, coming to the orthologous group analysis, we compared the protein sequences of Moringa oleifera with protein coding sequences of 11 other plants, and found that Moringa shares more similarity with Carica papaya and Theobroma cacao, while forming a separate clade with Arabidopsis thaliana and Brassica oleracea. Figure B, it shows the number of orthologs that is shared by Moringa oleifera with the various plants. So here also Moringa oleifera shares a maximum number of orthologous groups with Carica papaya. Now coming to simple sequence repeats, they are the most widespread molecular markers in plant genomes and are a popular choice for estimating genetic diversity and molecular breeding. So around 92,163 SSRs were identified in 594 out of the 915 contigs that we identified in the whole genome, and the tetranucleotide repeats were most abundant. Now coming to genic SSRs, they are helpful in assaying functional diversity and marker-assisted selection in plants. So we identified 3,362 SSRs in 31,056 protein coding genes, and here also trinucleotide repeats were most abundant, followed by tetranucleotide repeats. Now we went ahead to do the RNA-seq of control and drought-treated leaf and root tissues of Moringa, Bhagya variety. We isolated the RNA, went for library preparation, and then we did the sequencing. The RNA-seq generated about 753.5 million reads, which were filtered for quality, and these high-quality reads then were mapped onto the transcriptome of Moringa. this is a representative heat map of the differentially expressed genes under control versus drought conditions in leaf and root. So we also did a gene enrichment analysis for the DEGs in response to drought stress, and we found that most of the genes are enriched in biosynthesis of secondary metabolites and metabolic pathways. So based on the heat map and the pathway analysis, we identified key DEGs in the Moringa genome, such as zinc finger containing transcription factors, ethylene responsive transcription factors like WRKI, PHLH, transporters and genes associated with secondary metabolite production, and heat shock proteins and heat shock factors. So we selected heat shock factors to work on. The reason being that there are a few reports which suggest the role of HSFs in abiotic stress responses, and also it is linked to vitamin C biosynthesis. So we hypothesized that it might act as a potential regulator of antioxidant response in under drought stress. So the first step after that was to identify HSFs in Moringa oleifera genome. The HMM profile for HSF from PFAM and protein sequences from the plant transcription factor database was downloaded. BLAST-P was done to identify HSFs based on homology, and the redundancy was removed to get 21 unique HSF sequences that we named as small HSFs. Now figure A here, it shows the classification of HSFs into three classes, A, B, and C, based on homology with Arabidopsis HSFs. And figure B, it shows the gene structure of mol HSFs based on intron-exon composition. Now coming to molecular phylogenetic analysis, here we compared the HSFs of Moringa oleifera with the HSFs of 5 other plants. And here we can see that the mol HSFs share close similarity with the HSFs of Theobroma cacao, and whereas Oryza sativa is a clear outlier. This data further corroborated with our orthologous analysis data. Now, HSFs, they are known to bind to heat shock elements or HSCs in the promoter regions. So we identified the genes with HSCs in the promoter regions. Firstly, we extracted promoter sequences for all the CDS that is predicted in the Moringa genome, used a fine motif program to search for HSCs in the promoter region of all the sequences, and annotated the corresponding protein sequences using UniProt SwissProt database. So we identified a total of 1490 transcription factors containing heat shock elements in Moringa genome. Now, we also did the expression analysis study where we subjected the Moringa plant to drought by withholding the water for 10 days, and then the leaf and root tissues were collected for RNA isolation, and the expression analysis was done using QRT-PCR. Okay, so the QRT data showed that in case of leaf, there was downregulation of all the mol HSFs, whereas in case of root, there was significant upregulation of mol HSF8 under drought stress, and that is the reason we chose mol HSF8 as a candidate gene. Now, once we have selected our candidate gene, we are in the process to identify binding regions of mol HSF8 in the promoter regions through ChIP-seq, and then we will be validating it through Y1H or EMSA, and then we will be functionally characterizing mol HSF8 either through generation of knockout or overexpression. Additionally, we are also doing metabolite profiling to identify differential secondary metabolites in control versus drought stress condition, achieve our final goal where we'll get a improved drought tolerance with enhanced nutritive quality in crops. Now to summarize, we assembled about 90% of the Moringa oleifera genome into 900 contigs with an N50 value of 4.7 mp and predicted 32,062 putative protein coding genes. Based on the transcriptome data, we chose to study HSF family of genes to analyze their role in drought tolerance and antioxidant activity. A total of 21 HSFs in the Moringa oleifera genome were predicted. Genetic analysis, multi-identification analysis of gene duplication events, and differential expression of HSF coding genes were carried out. To conclude, the members of the HSF family have an important role in the plant's response to abiotic stress, and mol HSF8, since it is represented in the root system, it's a viable candidate for further characterization. These are the publications that I'm aware of, and I would like to thank my former guide, great Ajay Kumar Tatra, for anything and everything that he's done for us. My current PI, Professor Pulak Kumar Mukherjee, Dr. Seema Pradhan, Dr. Namisha Sharma, and Dr. Shubhobrat Majumdar for their constant support and guidance. AgriGenome Labs for assisting in the whole genome sequencing. Krishi Vigyan Kendra for providing us with the Bhagya variety seeds. RCB for registration, TST for my fellowship, my lab mates and friends. Thank you.
Thank you so much, PC Sri Shamli. Can I request Shagar Aditya? He is connected with us virtually. Right now, is he present? Can he present on the topic? Yeah, he's on, I guess. Okay, we have one more speaker with us, Mr. Sagar Aditya from National Institute of Technology, Agartala, India. He will be connecting with us virtually on a topic, utilization of different modes of predict tea crop yield, a critical review. Over to Sagar Aditya.
Thank you. வேற பிரச்சனை Number slide.
Hello. Sagar Aditya, can you hear me?
Hello.
Good morning, all. Am I audible?
Okay, all right. So we couldn't connect with Sagar Aditya virtually, but we have with
Sir,
us
madam,
Vanita
am I
Das.
audible?
She will be talking on the topic policy stress in Ethiopian A 5 UTR written the transcript in the
Shall I go ahead with my
University
presentation?
of Kalyani, India. Shanita.
Hello. 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 Ma'am.
Yeah.
Actually, Suganti ma'am is already present the screen. That's why I not present the screen right now. If she mute her screen, then I will present. Na? Actually, Suganti ma'am present her screen, so that's why I not able to my screen. Hello.
Yeah, sir. Hello.
Actually, Gopinathan ma'am present.
Yes, sir.
Shared your screen, so actually my name is there, so that's why if you not mute your screen, there I cannot able my screen share.
Okay. Okay, sir. Mute பண்றதா நான் ஷேர் பண்ணு.
அவங்க ஷேர் பண்றாங்க. PowerPoint புரியுது.
Hello, now my screen is visible, ma'am. Hello. Hello. Now, everyone. Good morning, all of you. My title is Utilization of Different Models to Predict Tea Crop Yield, a Critical Review. I am Department of Civil Engineering, NIT Agartala. My topic is, I already told you, utilization of different models to tea crop yield. So what is tea? We all know that, but tea is the most popular beverage in the world after water. It's a simple preparation of pouring hot water over cute leaves of the camellia plant. India is the second largest producer of tea globally after China. As a 2018 survey, a total of 6.37 lakh hectares of area was cultivated in India for tea production. Tea is the most important cash crop for Indian market. Over 15 nations manufacture it, with China, Kenya, Sri Lanka, Vietnam, Turkey, Indonesia, Iran serving the top exporter according to FAO 2014. There are mostly two types of commercial tea comprised of two different types. One is Assam type of tea and second one is China tea. Tea is an evergreen staff indigenous to northern India, cultivated in acidic soils from tropical to subtropical regions. It is one of the most valuable cash crops in India and an important source of different beverages which were claimed to be the most widely consumed fluids after water. It is an important beverage crop induced in India, initiated by the British government. Geographical condition of growth and production, the tea plant grows well in tropical humid and subtropical humid climates. India is a leading producer as well as exporter of tea in the world, where 28% of tea of the world is produced here. In India, Assam's hills of Darjeeling, Jalpaiguri, West Bengal, Tamil Nadu, Kerala. Apart from this, Himachal Pradesh, Uttarakhand, Meghalaya, Andhra Pradesh, and Tripura also tea production states in India. Here are some graphs: tea plantation area, total tea production, and tea yield. 1985 to 2015. Now, tea production and importance. The Chinese were the first to use tea as a medical drink, later as a beverage, and now serving as a morning drink of nearly two-thirds of world population daily. About 3 billion kilograms of tea is produced and consumed yearly. India was the top producer of tea for nearly a century, but was replaced by China as a top producer in the 21st century. While India is the largest consumer of tea worldwide, the per capita consumption of tea in India remains a modest 750 grams per person every year. Tea is consumed as part of daily diet to reduce the risk of cardiovascular disease and cancer also. Here some graph, world map of tea producing regions. The scenario of in Northeast India. So Robert Bruce and Charles Bruce were first to discover the tea drinking practice in Assam among the Shingpo tribe in the 1830s. Since 1940, Assam Tea Corporation (ATC) started commercial production of tea and became the leading producer of tea in the world. The country's tea production is declined by 1.3% to 966.4 million kilograms in 2010 due to output drop in Assam. The state produced 564,000 tons of tea per annum in 2007, while it came down to 460,000 tons per annum in 2010, as per report 2011 FAO. Here, northeastern India. Here the map northeastern India tea producing regions. Now, what is the geographical condition to growth tea and productions? The tea plant grows well in tropical humid and subtropical humid climates, endowed with deep and fertile, well-drained soils rich in humus and organic matter. 150 semi-summer rain and 21 degrees centigrade to 27 degrees centigrade daily temperature is needed for its cultivation. Tea bushes require a warm and frost-free climate all through the year. Tea is often cultivated on the hill slopes so that there is no waterlogging in its roots. Its leaves are to be plucked several times a year, so the availability of abundant and cheap labor is essential for its cultivation. Here's a literature review, the model's names and findings. So first, modeling the impact of agrometeorological variables on regional tea yield variability in South India tea growing regions. Authors Raj et al. What the method used? They use Ceramics, ANN model, Van model. Among them, statistical models and Van models produce the best fits for monthly, seasonal, and annual fits. On the use of statistical models, predict crop yield response to climate change, second title. Here, maize yields were simulated using increased maize method are used. Evaluate maize growth and yield response to change in management and environmental condition are findings. Some other titles are given. So machine learning approaches for crop yield prediction and nitrogen status estimation in precision agriculture, a review title. Method used machine learning. So what the findings? The findings, the rapid advancement approaches and sensing meteorology will offer complete cost-effective solution for improved crop and environmental status estimates and decision making. A survey on different data mining techniques for crop yield predictions. Their method use ANN, KNN, and SVM. What the findings? The finding using data mining techniques, the yield prediction problem can be resolved. Exploring the relationship between climate change and rice yield in Bangladesh and analysis of time series data. The method uses regression model and time series analysis. The ordinary last square and median regression approaches, we analyze time series data for 1972 through 2009. They use and climate variables rice yields. A hybrid approach to tree crop yield prediction using simulation models and machine learning. They use machine learning and aquacrop model. The findings, the aqua data from extra data from food database included soil agro-management data. Use of data mining technique for prediction of tea yield in the face of climate change of Assam in India. They use data mining techniques to estimate tea yield analysis. What the finding? The findings, crop production in response to the climate, such as rainfall, temperature, relative humidity, evaporation, sunshine hour, affect across the 4 tea growing regions, which were Kochur, Upper Assam, North Bank, and the small portion of Assam. India's tea price analysis is based on ARIMA model. They use ARIMA model. What is the finding? The purpose of this paper is built in ARIMA model to predict the price of Indian tea actions. So we utilize the model established above and use state relevant software to forecast Indian tea action price in the last week of 2014 and the first 2 weeks of 2015. Machine learning time series model to T-based hypothesis inflation in India. They use machine learning model. Evaluating the neural network model, accuracies can be done, the reevaluate data from the field. Yield prediction and waterlogging assessment for tea plantation land using satellite image-based techniques. The finding, this study was confined to a single vegetation type for prediction and assessment of waterlogging when the soil under has investigation, considered vegetation cover. Modeling shoot growth and yield of Ceylon tea cultivator. They use regression model. The findings, the model simulated leaf area and fresh weight of tea shoots at different development stages and location, which were good agreement with the measured values at radiation range R square. R square here shows 0.922 and 0.98 respectively. So according to the literature review, what are findings research gap? There is no through study to discuss how much does climatic variability influence for tea production. In this study, no proper modeling has been addressed for time series analysis on tea yield cultivation using multiple linear regression analysis. How much of the real world tea producing system vulnerability to climate change? Statistical analysis in tea yield not done yet with different atmospheric and weather parameters. Here my references are there. Thank you.
Hello. Thank you, Sagar Aditya. And once again, I would like to request Swensita Das.
Good morning. I feel myself blessed to be present and deliver my presentation amongst the August gathering here. I will take 15 minutes from your valuable time and request you to be by my side. The topic of my presentation is policy stretch in eutrophin A5' UTR retains the transcript in the nucleus. Duchenne muscular dystrophy is a fatal neuromuscular disorder that affects 1 in every 3,500 males worldwide, which leads to progressive muscle weakness and death of muscle tissue. The cause of Duchenne muscular dystrophy is mutation in dystrophin gene, and DMD is inherited in X-linked recessive manner. Usually, the symptoms appear around the age of 2 or 3 years. The characteristic features of this disease is calf muscle hypertrophy and curvature of spine. The patients face difficulty while arising from the floor, and they use a specific pattern for standing, which is called Goa's sign. Eutrophin is regarded as autosomal homologue of dystrophin due to its extensive sequence homology. Animal studies suggest upregulation of eutrophin can compensate dystrophin loss. Utrophin A mRNA undergoes extensive post-transcriptional regulation mediated by both 5' and 3' UTR region. Mouse utrophin A 5' UTR has a poly-C stretch which makes it possible target for poly-C binding protein 2 or PCP2. PCP2 has 3 cage domains and sequence-specific RNA binding capacity. There are several reported functions of PCP2 like replication control, translation and transcriptional regulation, and stability. To investigate if PCP2 interacts with 5' UTR of mouse utrophin A mRNA in C2C12 cells, we have performed RNA-IP using non-transfected cells. Upon using utrophin-specific primer, we have obtained 150 base pair RT-PCR product. Then cells were transfected with 5' loop construct which contains 507 nucleotide 5' UTR at the upstream of firefly luciferase open reading frame, and then RNA-IP was performed and we got 150 base pair RT-PCR product. But in case of serial construct which contains 5' UTR devoid of poly-C stretch, no RT-PCR product has been obtained. So it confirms that PCP2 interacts with 5' UTR of mouse utrophin A mRNA in C2C12 cells. To check if PCP2 modulates the expression of utrophin A mRNA, we have overexpressed PCP2 with the help of pcDNA PCP2 construct and silenced PCP2 with help of TBS U6 PSI construct in C2C12 cells. And it was observed upon overexpressing PCP2, the abundance of mRNA of utrophin was slightly increased, and upon silencing, it was slightly decreased. Surprisingly, in Western blot analysis, the utrophin protein level was slightly decreased by overexpressing PCP2 and increased twofold by silencing PCP2, which clearly confirms PCP2 plays crucial role in post-transcriptional regulation of utrophin expression. Then we want to check if the post-transcriptional regulation is mediated by the stability of utrophin A mRNA. So actinomycin D, which is a transcriptional repressor, was added to overexpressed and silenced cells, but the result shows PCP2 does not have any role in stability of utrophin A mRNA. Then we wanted to check if the post-transcriptional repression is mediated at the level of translation. So in vitro translation experiment was performed with reporter construct, and luciferase activity was slightly decreased in presence of PCP2 in the case of 5' loop construct. Then we wanted to recapitulate the effect of PCP2 in transfected cells. So the reporter construct was transfected in cells and no change in luciferase activity was observed. So the role of PCP2 in post-transcriptional repression of utrophin mRNA cannot be solely described in the terms of translation. Then we asked if it is nuclear event. Then fractionation was done, and the nuclear to cytosolic abundance of mRNA, of utrophin A mRNA, was 7-fold higher than beta-actin control, which confirms mouse utrophin A mRNA is retained within the nucleus. And RNA fish results also confirms this. The retention of utrophin A mRNA was increased upon PCP2 overexpressing and decreased upon PCP2 silence. nuclear detention of 5' loop construct was also increased upon PCB2 overexpression. Then the policy stretch of 5' uter was inserted at the upstream of firefly luciferase open reading frame, and the nuclear detention of this construct was more pronounced upon PCBP2 overexpression and decreased upon PCBP2 silencing, which confirms that the policy stretch at the 5' uter of mouse utrophin mRNA is responsible for the transcripts to be retained within the nucleus. This is RNA fish experiment results. The red puncta denotes utrophin mRNA here. The number of red puncta increases upon overexpressing PCP2 within the DAPI-stent nucleus compared to control set, and the number of puncta decreases upon PCP2 silencing compared to the control set. Then we want to check the status of utrophin in human. So we checked a number of cell lines, and we have found 590 base pair product, which is specific for utrophin 5' uter in 293 cell line. The utrophin protein level in this cell line was also checked by Western blotting, and then nuclear to cytosolic abundance was also checked. MALAT1 was positive control here, which was retained within the nucleus, and the nuclear cytosolic abundance of MALAT1 was more, and nuclear cytosolic abundance of utrophin was even more, which confirms the nuclear detention of utrophin mRNA in human cell line by PCP2. So we can conclude the nuclear detention of mRNA is a novel function of PCP2. In future, we aim to identify the nuclear detention elements in human and upregulate the utrophin expression through genome editing of nuclear detention elements. I am grateful to Dr. Utpal Basu, my guide, Department of Molecular Biology, Biotechnology, University of Kolkata, for his guidance and immense support. I would like to thank Dr. Gargi Ghosh, Thomas Jefferson University, Philadelphia, and I would like to thank all my lab mates. I am obliged to Dr. Jishu Nashkar, Dr. Arnab Gupta, Dr. Soumya Singh-Roy for their immense support. I would like to thank Dr. Shoran Das for his continuous inspiration and support. And especially, I would like to thank Society for Agricultural Research and Management, Cuttack, Odisha, for giving me the opportunity to present my work here. Thank you.
Thank you so much, Vandita Das. Next, we will be connecting virtually with A. Sugandhi ma'am. She is with us virtually. She will be talking on the topic ecotoxicity studies for Indian honeybees for Gangan and Moringa and mango. So she will be connecting with us virtually. She is from TNAU, Coimbatore, India.
Good morning, all. Am I audible?
Hello, Chick. Yes, ma'am, you're audible.
Yeah. Thank you. So my topic for today's presentation is ecotoxicity studies for Indian honeybees foraging in Moringa and mango. This is Sugandhi, Associate Professor from Tamil Nadu Agriculture University, Coimbatore. So this study was planned to assess the impact of selective insecticides that were applied on focal crops of Tamil Nadu, that is mainly mango and moringa, which are cultivated in the semi-arid regions. mango is cultivated for its fruits and moringa for its leaf, pods and flowers. And these two crops are benefited by honeybees, as well as honeybees are benefited by this crop. So the main objective of our study was to assess the lethal effect of the selective insecticides applied on mango and moringa, and then to study the impact of these insecticides on the colony performance, brood performance, and the food reserves of the Indian bee Apis cerana indica, and then to quantify the insecticide residues and assess the risk of these insecticides to Indian bees. So coming to the methodology, we conducted bioassay studies in the laboratory for assessing the lethal toxicity of the insecticides. In our study, we selected around 22 insecticides based on the preliminary survey conducted among mango and moringa farmers. So the LC50 studies were conducted in the laboratory for these 22 insecticides. Sugar solution was prepared with the insecticides, selective insecticides, and this was fed to the honeybees. So from the picture, you can see we used plastic cups of 6 centimeter dia, and the lid carried a vent through which the feeders with sugar solution contaminated with insecticides were given to the honeybees. The mortality of the honeybees were recorded after 6 hours, 24 hours, and 48 hours. So with a suitable range and series of concentrations, we did probit analysis following Finney's method. And by this way, we fixed the L60 dose, that is the dose that gives 50% mortality of the honeybees. So this way, the L60 was arrived for all the 22 insecticides. From the table, you can see this, out of the 22 insecticides we tested, clothianidin topped the list with very high toxicity. The lethal concentration 50, LC50 value was 0.0015, and this was followed by fipronil, thiamethoxam, imidacloprid, and dinacloride. So these 5 insecticides topped the list. And then we also assessed the LD50 values, that is lethal dose for all these 22 insecticides. For the lethal dose calculation, the insecticide solutions were applied on the thoracic region through a micro applicator. The thoracic region is the more suitable exposure site for honeybees. So we did topical application with the micro applicator. You can see in this picture. 1 microliter of the prepared insecticide solution was applied to the honeybees through the thoracic region. And again, the bee mortality was recorded after 6, 24, and 48 hours. And if the insects were live, again, we made observation further to 48 hours also. Then the second part, we did field experiment in moringa and mango crop. Open field studies and semi-field studies were conducted. For semi-field studies, the cages were made with insect-proof nylon mesh of size 10 meter into 6 meter into 3.5 meter width. So this tunnel was made and erected using cashew poles or iron poles. And then both in open field and semi-field conditions, we maintained 3 Indian beehive, that is Apis cerana indica hive. These hives were placed inside the semi-field as well as in the open field 2 days before the application of insecticide. And the application of insecticide was taken during 20% blooming of the crop. So that is the right stage when the forages, honeybee forages will make more visit. So we preferred that time, and the field experiments were implemented. Then the third part of our research was an residue analysis of the selected 22 insecticides in honeybees. So whenever honeybees go out for foraging, they feed the pollen and nectar, and through this process of the residues, they enter the honeybees. So the persistence of these residues in honeybees were analyzed using liquid chromatography, mass spectrometry technique. So similar to oral LC50 values, the LD50 values were also studied for Indian honeybees. Again, here clothianidin, fipronil, and thiamethoxam topped the list. For clothianidin, fipronil, thiamethoxam, imidacloprid, the toxicity was similar to LC50, but next to this, all the other insecticides, there was slight variation in the toxicity order with respect to the route of exposure. So the thiacloprid was found to be the safest among the 22 insecticides tested comparatively, because it had higher LC50 value as well as LD50 dose. So these figures shows these pictures shows our field experiment conducted in Moringa. Here you can see the semi-field experiment condition where the cages were erected and the hives were maintained. In front of the hive, we placed a deadbeat trap. This was placed just below the entrance plate. So whichever honeybees are exposed to pesticides, and when they return back, so they'll just lie in the front and they'll die. So such dead bees were collected using the dead bee trap. And in this right side picture, you can see the dead bees that were collected after exposure to the applied insecticide. Yes, now from this table, it is visible that the fipronil, insecticide fipronil, is more toxic among the different insecticides tested in Moringa. Moringa, for Moringa, there is no recommended insecticide or approved insecticide by Central Insecticide Board Registration Committee, but we have selected this insecticide based on farmers' practice. So farmers were using imidacloprid, fipronil, chlorantraniliprole, imamectin benzoate, and thiamethoxam. All these insecticides were compared with the standard check dimethoate. So among these insecticide tests, fipronil was found more toxic to honeybees. The population, it reduced from around 3,000 to nearly 100 after 15 days after application of the pesticide. So followed by fipronil, the next toxic chemical was thiamethoxam, and this was followed by imidacloprid, chlorantraniliprole, and chlorantraniliprole. The ivermectin group of insecticide, that is imamectin benzoate, was found safer than the other groups, and this was almost on par with dimethoate, the standard check. The impact of these test insecticides were also studied with respect to brood area. So brood area, it means the egg, larva, and pupal population. So again, so the observation were taken on 3rd day, 7th day, 20th day, and 28 days after application of the insecticide, selective insecticide in Moringa crop. Here again, fipronil proved to be very toxic, highly toxic with reduced brood area. So here we measured the brood area using a 1 into 1 centimeter grid frame made of a polythene sheet, and this was placed again each live hive, and then we made the observation. So again here fipronil proved highly toxic. The brood area gradually reduced. This was followed by thiamethoxam, imidacloprid, and chlorantraniliprole. Imamectin benzoate proved a bit safer when compared to the other insecticides. This is the field experiment in mango. Here again, you can see the Indian beehive that is placed in a full bloom mango crop. And in front of the hive, there is a dead bee trap. This picture shows the semi-field experimental condition. And yeah, here you can see some the honeybees collected in the dead bee trap. And this is the picture where you can see the grid that is placed against the hive with bees, just to count the brood area, food reserves like nectar store and pollen store. This is how observation was taken after exposing the hives to insecticide applied crop. Now, from this table, we can see the impact of insecticides applied in mango to honeybees. So this data is taken from semi-field trial plots. Here in mango, thiamethoxam proved to have very significant effect on the colony population. There was more mortality with when the crop was applied with the thiamethoxam 25WG, and this was followed by imidacloprid, then lambda cyclothrone, that is a synthetic pyrethroid again, and this was followed by torfenpyrad and the standard check dimethoate. So again here the neonicotinoid group of insecticides, thiamethoxam and imidacloprid, proved highly toxic to honeybees when compared to the newer molecule, that is pyrazole insecticide torfenpyrad. All these 5 insecticides are recommended or approved by Central Insecticide Board and Registration Committee for use in mango crop for management of different insect pests. So again, the impact of these test insecticides in mango were studied for their impact on brood area. Thiamethoxam proved significantly toxic, and it had a very adverse effect on the brood area development. This was followed by imidacloprid, lambda cyclothrone, torfenpyrad, and then dimethoate. So all these were significantly different when compared with the control treatment, means the hives that were unexposed to any pesticide application. Then coming to the third part of our research, we did a pesticide residue analysis in bees. So just to know the level of contamination in bees. So the residue analysis was done using liquid chromatography, mass spectrometry technique. We used water and astronitrile as the mobile phase, and this was run as a gradient program. All the 22 analytes were analyzed both in ESA positive, that is electrospray ionization in positive and negative mode. For each analyte, both the parent and 2 daughter ions were monitored. And based on this, confirmation and quantification was done. So multiple reaction monitoring transition was done for all the 22 analytes. Then coming to the sample analysis, 5 gram of honeybee sample was used. So in Indian bee honey, the average weight was around 17 milligrams. So for 5 gram bee sample, we had around 150 to 200 bees. So this 5 gram of bee sample was homogenized. This was ground. This was added with the astronitrile solvent and NHN. Usually astronitrile, it is the polar solvent used for extraction of residues. In our methodology, we used 2 ml of NHN. This was mainly because honey bees, they contain wax and lipids. So just to remove those wax and lipids, NHN was used. So after the extraction of the residues from the matrix, the sample was cleaned using salts, magnesium sulfate, primary, secondary amine, and rubberized carbon black. After cleaning all the interfering material from the solvent extract, the solvent was further added with the acetonitrile, and then this was spread in LC-MS for quantification. So here in this picture, you can see the standard chromatogram. This was arrived after injecting the B matrix that was added with the standard mix of pesticides, and this chromatogram was obtained after feeding it in the LC-MS instrument. So here I have highlighted the 2 ions for each analyte. For example, this is the chromatogram corresponding to imidacloprid. So here 2 ions, one for quantification and one for confirmation. Both ions were monitored. So likewise, for all the 22 insecticides, we monitored both the qualifier ion and quantifier ion just for confirmation. So next, after developing this method, which was validated, the recovery studies and precision studies were done. So we achieved the recovery between 70 to 120%, and the precision was below the acceptable criteria for relative standard deviation below 20%. So with this validated method, we went for analyzing the field samples. The dead bees that were collected from the sprayed moringa field and the mango field, they were analyzed. In case of moringa, the bees collected from chlorantraniliprole treated field showed residues up to 10 days. Here you can see the residue level was very high during the 3rd day observation. The 3rd day sample contained around 7.17 ppm of chlorantraniliprole, and the residues persisted until 10 days. Though fipronil was very toxic to bees in moringa crop, the persistence was very higher in chlorantraniliprole. This was followed by thiamethoxam, imidacloprid, and fipronil. Imamectin benzoate, it persisted for short time when compared to chlorantraniliprole, thiamethoxam, or fipronil. And no residues of dimethoate were detected in the bees. They were below the quantification limit set by our laboratory. So this proves that some chemical have a longer persistent effect, though they are less toxic. And some which are highly toxic, they persist for less time. This is again the results of residue from the bees that were collected from mango moringa field. Here, yeah, this is from mango field. Sorry. Here again, thiamethoxam and imidacloprid persisted until 7 days, and no residues of tolfenpyrad and dimethoate were detected in any of the dead bees. So these studies were conducted both in semi-field and open field condition. Then we did a hazard analysis based on the exposure of honeybees. So this we studied by simulation method. The honeybees were placed in a cage, and then they were sprayed with insecticide solution. So from the picture, you can see there were tiny droplets that settled on the honeybees. So whenever the forages go to the bee, they get exposed to the pesticide sprays. So such a situation was simulated here. Then the pesticide droplets were measured using light microscope. Thus, the volume was arrived. based on the concentration volume of the droplet, the residue that deposited on each bee was calculated, and the hazard quotient was analyzed by dividing the concentration and volume of the droplet by the contact LD50 values. Here, the LD50 value is more, the hazard analysis index was more than 1% for all the 4 insecticide tested. So this indicates high risk of these chemicals. If the hazard quotient value is between 0.1 and 1, then that indicates moderate risk of the insecticides. And if the hazard quotient value is less than 0.1, then that indicates low risk. But in our case, all the 4 insecticides which were tested in moringa or mango, they were found to be highly risky to the honeybees. Then we analyzed some of the probiotic bacteria that were analyzed, that were isolated from the gut of honeybees. So we could find Enterobacter species and Bacillus species from the gut of honeybees. There were several reports from Apis mellifera that these microbes play a role in increasing the immunity of the honeybees. Here again, when Bacillus subtilis was mixed with the pollen supplement of honeybee, and when it was fed to the honeybees, the LC50 value, that is the lethal concentration value of fipronil, flunicamide, dinotfuran, imatinib benzoate, tolfenpyrad, and dimethoate had higher LC50 value, indicating reduced toxicity. So some of though the function of this probiotic bacteria Bacillus subtilis is not clearly known, it plays a role in reducing the toxicity of the insecticide, or in other way, it increases the immunity of the insecticide. So this again opens a new avenue in the research area for honeybees, where we can put more work on identifying the probiotic bacteria that can be used in the pesticide residue mitigation of honeybees. So finally, I conclude that more research on the exposure of Asian honeybees and other domesticated bees and sting bees need to be strengthened. So in many cases, all the studies are based on Italian honeybee, that is Apis mellifera, and all those findings were extrapolated to Asian honeybees. So this we have to strengthen our research more in Asian honeybees specific to our Indian crops. Then cross-specific recommendation of the insecticides has to be given to farmers to harness the benefit of honeybees. Then the significant risk of sublethal doses of insecticides need to be studied to Indian bees, because some insecticides, though they are very less toxic, they persist for a long time in the environment as well as in the bees. So such sublethal effects need to be studied. Probiotic microorganisms need to be included for mitigating the impact of pesticide residues in honeybees. Thank you. Thank you for the opportunity given. So this project is funded by National Bee Board. So I take this opportunity to thank the National Bee Board New Delhi for providing us the grant. Thank you.
Thank you so much, Sugandhi ma'am.
Yeah.
Next, I would like to request with us here Ms. Shrestha Devnath. She will be talking on the topic, mangrove algae could be a one-pod platform for cellulosic waste, recycling, and biodiesel liquid generation. So she is from JIS University, Kolkata, India. Everybody requesting you to please put your hands together for Ms. Devnath.
Apa? Leveling.
বলছি।
So good afternoon everyone. I am Shrestha Denna, University, Kolkata, Department of Biotechnology, under guidance of Dr. Dimakar Ghosh from Microbial Engineering and Algal Biotechnology Laboratory. So today my presentation topic is mangrove algae as a sustainable microbial cell factory for cellulosic biomass degradation and lipid production. So human civilization faces huge energy crisis due to climate alteration and reduction in fossil fuel reservoir. So my target is biofuel extraction from mangrove algae and as well as diesel production and cellulose degradation. So lignocellulose is accumulation has been increasing through anthropogenic activities. Few studies show algal endonuclease and cellulase activities for degrading lignocellulosic waste to generate monomeric constituent which can further be channelized to produce higher biomass and lipid content at biodiesel precursor. Thus, the present study focuses on the feasibility attempt to show that mangrove algal isolate could be a potential microbial cell factory for cellulose degradation as well as the higher lipid production. So my aim and objective is the lipid production and cellulose degradation from mangrove algal isolate. At first, the materials and method, I have cultivated and microscopic observation on BS3 and BS4 algal isolate. After that, I determine the growth and the lipid content in BS3 and BS4 algal isolate. After that, the qualitative and quantitative assessment on cellulose degradation potentiality of BS3 and BS4 algal isolate I have done after the studies on phylogenetic analysis of algal cellulose and microscopic observation of BS3 and BS4 algal isolate. The first material and method is cultivation and microscopic observation on BS3 and BS4 algal isolate. Experimental study had been initiated through collected the water sample from mangrove ecosystem in Sundarban Island. Afterward, 2 ml river water sample has been filtered and it sterile and growth in BBM basal bolt algal medium. After that, it was incubated in 16 watt per meter light intensity shaking mode for 7 days. After 7 days of incubation period, the green color appears in the culture tube. And this is the sample collection picture from Sundarban mangrove ecosystem. After that, the determination of growth and lipid content in BS3 and BS4 algal isolate I have done. The growth profile of novel algal isolate had been determined following spectrophotometric approach by preparing the standard curve between optical density and biomass. And in vivo qualitative estimation of lipid droplet or lipid accumulation had been determined by the classical Sudan black staining method. After that, I have done the qualitative and quantitative assessment on cellulose degradation potentiality of BS3 and BS4 algal isolate. The confirmation of cellulose degradability had been initiated through cellulose Congo red assay, where the selfie supernatant had been used for in vivo degradation assay, including CMC as a carboxy methyl cellulose as sole carbon substrate with the concentration of 1% weight by volume. After that, I have done the studies on phylogenetic analysis and of algal cellulose and microscopic observation of BS3 and BS4 algal isolate. And the phylogenetic trees has been drawn using the neighbor joining method. And however, the main objective doing this phylogenetic analysis was to classify the diverse ranges of algal cellulase or endoglucanase enzyme. So this is a result of screening and cultivation of pure algal culture. These are the algal isolate cultivation and maintenance. After the microscopic view of algal isolate at 400x and 1000x magnification, the microscopic observation of mangrove ecosystem derived algal isolate. Here are the lipid droplet, which are blue color shows. This is the phylogenetic trees has generated based on putative algal cell 9B and cell 9C protein sequences. This is the comparative overview on characterization of BS3 and BS4 algal isolate through microscopic observation and phylogenetic analysis. And this is the BS4 algal isolate, which is the potential algal isolate which has the extracellular cellulose activity has been present, and this is the profile of secretion of cellulitic enzyme of BS3 and BS4 algal isolate, which is using the carboxy methyl cellulose supplementation and Congo red plate assay. This is a dye decolorization assay which has been done before and after treatment with and without Congo red to 0 to 9 days. And this is the decolorization activity which was the highest for BS4 algal isolate, which was the 96.04%. This is the DNase assay I have done for sugar estimation for the measurement on reducing sugar release after cellulose degradation using algal isolate BS4. After treatment, this is the after treatment for BS4 shows that the amount of released reducing sugar high. This is the deep brown color after treatment, and this is the reducing sugar released sugar concentration graph for 0 to 9 days. This is the BS3 algal isolate, which is the BS3 isolate growth curve where the day-wise the biomass, OD, and optical density and the lipid content increases, and the extracted lipid has dried white palette here. This is the BS4 algal isolate, and this is the growth curve of BS4 algal isolate where the day-wise biomass, OD, lipid content increases, the extracted lipid as derived the white pellet. And this is my published research work about this research. And my future outlook is beside the cell 9B and the cell 9C, after I will do the try to cellulose degradant cell 9D module needs to be explored and understand the more efficiency of BS4 algal isolate. So here are the references. And I would like to thank JIS University Kolkata and JIS Group of Educational Initiative and Dr. Dipankar Ghosh, my PhD supervisor, and all my MIB lab mates. Thank you.
Thank you so much, Shrista. Thank you. Next, isolation on the topic isolation and characterization of phosphate soluble bacterial ingredient to improve soil fertility and plant growth by Palash Kurai. And he's from JIS University, Kolkata.
morning, everyone. I am Palas Ghodai. Mm, I'm PhD student of JIS University under Dr. Dipankar Ghosh super guiding. My topic is isolation and characterization of phosphate solubilizing bacteria inoculates to improving soil fertility and plant growth. Phosphate is a second most significant primary macronutrient in plant. It role play in plant growth, seed germination, and it formation of various type of macromolecules and micromolecules in plant like DNA, RNA, and ATP. In soil, phosphate insoluble form present in various amount, but this insoluble phosphate do not access plant directly. Phosphate solubilizing bacteria, these insoluble phosphate soluble by organic acid production and phosphatase enzyme production and formation the phosphate solubilizing and formation the plant accessible orthophosphate like monobasic phosphate and dibasic phosphate. This is the research gap. In current scenario, excessive amount of the chemical fertilizer use affect the soil fertility and long-term adverse effect in plant growth and food productivity. Biofertilizer is the most prominent solution. The, chemical fertilizer. And secondly, the in current scenario, phosphate solubilizing bacteria available in market, but do not fulfill the practical goal like low capacity of growth, functional efficiency like phosphatase enzyme activity, organic acid synthesis, and low stress tolerance capacity like soil, alkaline soil and acidic soil, and low survivability in diverse ecosystem. This is my objectives. Screening and isolation of phosphate solubilizing bacteria from mangrove soil ecosystem, and secondly characterization of this bacteria isolation, and third study of its application in rhizospheric stress soil for improving plant growth. This is the research workflow. First, isolate the phosphate solubilizing bacteria from mangrove ecosystem, Sundarban. Because mangrove ecosystem is a anoxic condition is very high and salinity condition is very high, and phosphate content is very high, and then identify the most potential phosphate solubilizing bacteria potentiality aspect like orthophosphate production quantification and phosphatase enzyme quantification, and then started the salinity tolerance study for commercialization standpoint, and inoculate the best phosphate solubilizing bacteria inoculate in saline rich acidic soil, and then study the plant root, shoot, and leaf growth. In this study, fulfill my two research goal. This is the improving the industrial and agriculture network and minimizing the chemical fertilizer application to improving sustainable agriculture. First, soil sample collect from Sundarban region, three region, that is Dobanki, Pakhilaloi, and Vidya Mandir, and then isolation of phosphate solubilizing bacteria, selective media, by selective media, PVK media. This PVK media contain insoluble phosphate, calcium phosphate, and this calcium phosphate soluble by phosphate solubilizing bacteria, and create a hollow zone, and isolate the 6 single cell colony, and these 6 single cell colony nomenclature by P1, P2, P3, P4, P5, and P6, and then quality ability cross check by oil diffusion method. This is the comparison. This picture is the comparison of phosphate ability, phosphate solubilizing bacterial ability by or inorganic phosphate solubilization diameter. This is the biochemical quantification of phosphatase enzyme by ascorbic acid method. This ascorbic acid method, uh, orthophosphate react with ammonium molybdate and produce blue color. These pictures show the comparative study of orthophosphate production at 5 days treatment in PBK media, and figure C, measurement of the pH change by PBK media treatment at 5 days, and phosphatase enzyme activity test by phosphate solubilizing microbes produce phosphatase enzyme, and this phosphatase enzyme react with paranitrophenol phosphate and produce paranitrophenol, and react with NaOH and produce water soluble yellow color formation. This is the quantification of the phosphatase enzyme activity at 5 days treatment in PBK media. This organic acid solubilization for use the lecithin for organic acid. Organic phosphate. And the morphology characterization based potassium phosphate solubilizing bacteria P5. This P5 bacteria is gram staining gram positive and rod shaped bacteria. And then phosphate solubilizing bacteria halotolerance efficiency study, commercial expectation. This in this the satisfactory result for 75 gram per liter NaCl. And then green chili plant growth study in acidic saline soil. For soil sample collection, Potaspur area from West Bengal, and then select the green chili plant for experiment. Green chili plant because over a long period of time, green chili plant have been growing in many location in take advantage, and this plant grow in garden whole region, whole section in West Bengal. After that, the show a single green chili plant growth of phosphate solubilizing bacteria treatment at 2 months. This treatment should height growth 10% and increase, and root length growth 10% increase, and number of branches of root growth up to 100%, and leaf height growth 25%. Conclusion and future outlook. In this study clear the present P5 bacteria isolate have been most capable for highly salt tolerance ability, and this study demonstrate the shoot and root morphology of plant green chili plant seeding are the significant effect by P5 bacterial isolation treatment in saline and alkaline soil. And future expectation molecular characterization of P5 bacteria, P5 phosphate solubilizing bacteria, and pathogenicity test for human health hazard, and viability study for commercial expectation. Thank you, JIS University. Thank you, my guide, Dr. Dipankar Ghosh, and MAAB lab mates. This is my some publications related this research. Thank you.
Thank you so much. Next, on a topic, application of lignocellulosic bacteria, consultate on jute crop waste processing and valorizations. I request Mr. Somnath Das.
Hello. Hello everyone, good afternoon. I am Shomnath Dash, PhD scholar of Dr. Dipankar Ghosh, Department of Biotechnology, GIS University. Today I am going to discuss about the application of lignocellulosic bacterial consortia on jute crop waste processing and valorization. So first of all, I would like to provide some news about the jute in recent Times of India newspaper. That is the Federation of Indian Export Organization has set an export target of $50 billion from West Bengal. Said the FICO Director General and CEO Ajay. That means that company is going to provide approx $50 billion to the jute industry in recent years. And the second thing is how American shoppers are changing West Bengal's future fortune. That means in America, basically what they do, they don't use the plastic bags. Rather, they would like to use the jute bags. So that is the thing. So they are changing their mind and they are basically using jute bags without using any plastic bags. And it is also in our India also, in every state, state government has said that we should use the jute bags without using the plastic bags. So we have to ban the plastic bags. So that is why the jute bags or any kind of jute product is the future. And the third thing is in West Bengal, empowerment through skill development and job creation. So as the importance of jute is getting higher day by day, so the jute industries are also getting exposed, and the new jute industries are growing day by day. And so new thousand of job creations will be there in near future. So from this scenario, from this newspaper article, we can say that the jute fiber, that means the jute industry is a growing industry, and it will take its position within 10 years. So these are some statistical analysis. In the first statistical analysis, you can see that the jute goods consumption in India from financial year 2009 to 2022. We can see the graphs due to the pandemic, corona pandemic in 2020, 2021 was not that much great. But again, as we are in the post-COVID area, so the new jute industries are getting higher day by day. So the jute consumption is also getting higher day by day. And in the second case, if we consider the jute export mechanism in India, we can see that in last 2015 to 2022, it is getting higher. It is getting higher day by day. So that means we can assume that what kind of impact jute industry has in India, because it earns millions and millions. So that is why this industry is very much important in nowadays and will be more important in future days. So these are some jute products that we generally used in our daily life. So first of all, we basically change the plastic sacks and the jute sacks will be there in future again. And we use the ropes, we use the bags, we use the hats and daily usable things. So that is why this is very important that that my main, I would like to say that how important the jute is and how can we use the jute in various aspects. So that was the good thing about the jute, but there are some drawbacks are also there. So if we can consider a jute plant, then we can see that we can divide it in three different sections. One is the tip portion, one is the middle portion, and one is the root portion. So my main aim and my main objective of my paper about the root section. What is the main drawback in jute industry? They basically cut the root section to get a good quality jute fiber. So if we can see that, you can see that the jute fiber, the jute industry produce will contain 5% of jute roots, in 10% of jute root, 15% on jute root. Or if you want to get a good fiber quality jute, then you have to cut the root portion. because the root portion of the jute plant is highly tough and is a highly tough lignocellulosic biomass. So they generally cut it down and they generally thrash it into the garbage. So basically there is no use of the root. But what is the main problem? As they cut the root portion, as we can see in the picture, the root portion is a 1/3 of the total plant tree. So if you cut the 1/3 of the jute plant, then I mean, if you don't use that portion, this is basically the economical loss for the industry, for the country, for the economy also. And the another thing is that if the root portion, which is a lignocellulosic biomass, if you dump it in the dump yard, this will form some lignocellulosic waste, this will form some toxic material. So that is basically harmful for the environment. So we can think in two different prospects, that one is the environment is getting damaged if we cut the root section and if we trash it into the dump yard. And the second is that if we cut the root section, as this is the 1/3 portion of the total jute plant, jute system, so we basically waste the fiber. We can get fiber from it. If we can recycle it, or if we can basically rate it properly by proper channel, then we can use the root section. You can use the root section, we can make different products from this jute plant root section. So, and that will obviously help in the economical growth in India. So that is the root section that I mentioned here. So that is the aim and object. There are different type of techniques we can use to use the root section in different way. That is the physical technique, chemical technique, physico-chemical technique, and the biological technique. But in physical technique, chemical technique, the root section getting ores. I mean, if you apply the physical technique or chemical technique, the root section getting more ores. So that is why we don't generally prefer the physical or chemical technique. Rather, we would like to use the biological technique. And if we consider the biological technique, we can consider in two different ways. One is the monoculture technique, and the second one is the consortia technique. Monoculture technique means when you are using a single microbial culture to rate the jute. And the consortia technique is that if you use more than one bacterial culture or any kind of microbial culture at a time, so it has a different effect. That means the consortia is basically provide faster rating time, and the consortia will provide the good quality of the fiber portion. That means it can rate the root portion very well, the consortia technique. But the monocultural technique is nowadays it is a old technique, so we would like to emphasize on the consortia technique. So here I already mentioned the different 9 to 10 plus point of using the consortia approach. So this is the methods and methodology. So what we basically did, we took the sample, we isolate the microbial system from Sundarban mangrove ecosystem, and in 3 different sections from Dobanki, Pakiraloy, and Vidya Mandir. And we screen them for the production of 3 different enzymes, that is the lacase, cellulase, and pectinase. As we know that the jute is nothing but a lignocellulosic biomass, so if we want to rate it, we have to use 3 different type of enzymes, that is the lacase, cellulase, and pectinase. And because the lacase helps to break the lignin, the outermost portion of the lignocellulosic biomass, cellulase helps to break the cellulose to provide different 5 and 6 carbon sugars, and pectinase is the cementing material. So if we want to basically modify the fiber system, if we want to basically rate the jute root section, we have to break the pectin portion. We have to break the mainly lignin portion mainly, because lignin is the top portion, and if we can break the lignin in the jute fiber, then the fiber quality will be enhanced. So that is my main goal. So what we did, we generally isolate 3 different lignocellulosic enzyme producing strain, that is lacase, cellulase, and pectinase, and we have screened them in different way by the enzymatic assay, by plate assay, by didecolorizing assay, qualitative and quantitative assay. And after that, we mix them in 4 different ways. That means a lacase producing bacterial strain will be mixed with the cellulase producing bacterial strain, and that is the 1+2. And in same way, 2 plus 3, that is the cellulase producing microbial system, will be added up with the pectinus producing bacterial strain. And then again 1 plus 3, that is the lacase producing bacterial strain plus pectinus producing bacterial strain. And in the fourth case, 1 plus 2 plus 3, that means the lacase plus cellulase plus pectinus in all in a single consortia. So this is basically the consortia, and we can grow it in different formulated medium and also in the nutrient broth. And after growing them in different system, different medium, we basically use them for the jute rating purpose. And after jute rating, we generally checked that whether they properly rate the jute or not. But for that, we analyzed the reducing sugar estimation. We did the FTIR analysis and also the same analysis. So this is the result section. So you can see from the picture, this is the qualitative as well as the quantitative method. So in the plate assay, we can see the lacase produce the halozone basically around the methylene blue plate. And in case of cellulase, it produces the halozone around the malachite green plate. And in case of the pectinase, it produces the halozone in the Congo red plate. Again, we can see we did the same mechanism in liquid media also, and you can check that from 0 hour to 6 hours change, the color become fade. So that means the microbial system are able to produce the enzymes that are required for the decolorization of that media, of that color basically. And why we did that decolorization assay? Because these the enzymes that we require for the breakdown of the lignocellulosic biomass is able to break this color. That means lacase can break the malachite green and the methylene blue. Samely, cellulase also break the Congo red or the malachite green methylene blue. So that is why basically check that whether this enzyme can break this color or not, and we get the positive result. And from that we can conclude that yes, these enzymes, and obviously this the plate that we made at the selective plate. That is why it is pretty clear from the picture that in selective plate the microorganism will obviously provide that enzyme that is properly for that medium. That means in the first plate we can see that is the lacase plate, that means this is the lignin plate. So the microbial system basically provide the lacase. That is why it breaks the color, and it makes the halozone there. And we did the same thing in the liquid media also. So this is the release reducing sugar versus biomass. That means we checked it for 21 days, from 0 to 21 days. And we check that the biomass is getting increased and the reducing sugar is also getting increased. So from that we can conclude that they are providing they are basically breaking the fiber, and they provide the sugar from that. Sorry. Okay, so this is the FTIR analysis of the consortia. That means we use 4 different consortia, and after using them for rating the root fiber basically, jute root fiber, we get this FTIR data. And from this data, we can conclude that the 1 plus 2 plus 3 system, that means the system having all the 3 required enzymes, provide the best result among all the other results. And this is the same scanning electron microscopy result. From this result, you can conclude that how much these enzymes are working in the root section, and they are able to break the lignin part, the tough part, the surface part of the jute fiber. And that is our goal basically, to break this lignin part, to make it more smooth, to make this more upgradation of the root section. So we became successful in this respect. And this is the future outlook. So I make this diagram to provide a short future outlook. That is, if we use the jute root cutting isolation, if we use the jute root, then we can, if we don't dump it, then we can do the lignocellulosic waste remediation. That means there will not harm for the environment. And second, if we use that jute root for the rating purpose by the consortium, we can upgrade the root fiber portion. And if we can upgrade the root fiber portion, we can use the fiber from the root section for the generation of different kind of value-added products. That means the valorization of the products. And if we can make the value-added products from the jute root section, we can use them to export in different countries throughout the world. And through this, we can help in the growth of our economy of India. So that is the important thing. So I would like to acknowledge my supervisor, Dr. Ghosh, and my MEOB lab mates, and the GIS University, and also my family for helping me always. And so these are some publications from our lab regarding this, research work. Thank you, everyone.
Hello, I'm Akanksha Yadav.
Yes.
I want to present my PPT.
May I know your name, ma'am, please?
Akanksha Yadav.
It's Rosin Raj, sorry. Akanksha Yadav, okay. Thank you so much, Somnath Das. It was really lovely. And I would like to request right now, she is connected with us virtually, Mrs. Akanksha Yadav, and she will be talking on the topic, identifications of integrated farming system in Vidyapur district of Rajasthan, a field survey. And she is from Maharana Pratap University of Agriculture and Technology, Udaipur, India. Next to Akanksha Yadav, she will be with us virtually. Ma'am, please turn on your camera.
Ma'am, I'm sharing.
Ma'am, share your camera and your screen too. Turn on your camera.
Ma'am, please give me 1 minute.
Do we have anyone offline here? No? Okay. After Akanksha, I would like to request Mr. Roshan Raj, if he is present.
Yes, ma'am, I'm present.
Okay. Akanksha, will you take more few minutes, or should I allow Roshan Raj? Is it okay with you?
Ma'am, please give me 2 minutes, please, ma'am.
Okay.
Ma'am, is it visible?
Yeah.
Good afternoon, everyone. I'm Akanksha Yadav, PhD research scholar from CCAS, Maharana Pratap Agriculture University, Udaipur. And my research topic is identification of existing integrated farming system in Udaipur district of Rajasthan. a field survey. And nowadays, agriculture is the most important part of the economy in India as it provides foods and livelihood security. It's play a vital role in the Indian economy and contributes 22.2% to the total GDP of in 2020 to 21. Currently, India has a second largest population in the world, and to meet the future demands, we have to produce 341 million tons of food grains and also provide employment to more than 60% of the population. Rajasthan state occupies nearly 10.4% geographical area of the country. In the state, agriculture is the main occupation in of the people. Among almost 70% of the population is engaged in agriculture and allied pursuits for their livelihood. The agriculture is the most part of the state in rain-fed and prone to high production risk. Presently, the state agriculture is facing many crises such as dry climate, droughts, famine, hailstone, pests and diseases, poverty among farmers, independence, and etc. Farmers' socioeconomic life and directly affected from agrarian classes. The integrated farming system is an integrative whole farm approach that effective in solving the problem of small and marginal farmer. The main aim of integrated farming system to enhance the employment and income from small holding by integrated various farm enterprises and recycling crops residue or byproducts within farm itself. The farmers needs to be settled of regular income for living at least above poverty line according to Dashora and Singh. And these are some benefits of the integrated farming system. The integrated farming system are an, an eco-friendly techniques in which waste from one farm becomes input and another, allowing for more effective use of farm resources. In the agriculture domain, the IFS can improve the following to improve the soil health and fertility, to also enhance the livelihood as well as status of the farm families, and to develop the multiple sources of income, to minimize the production cost and farm inputs requirements, also to reduce the use of pesticide and chemical fertilizer, and to enhance the economic yield per unit area, to develop the employment for the member and others, and also to effective utilization of the resources, to develop the balanced diet and nutrition food for the farm families. To next one is to resolve the problem of energy and biogas or solar system, also to avoid the degradation of forest and ponds. And next one is recycling the available resources. And my objective of the study is to identification of the existing integrated farming system in Udaipur district of Rajasthan. And my research methodology, the present study will be conducted in Udaipur district of Rajasthan. There are 20 Panchayat Samiti in Udaipur district. Out of 20 Panchayat Samiti, 4 Panchayat Samiti will be selected randomly for the study. And for each selected Panchayat Samiti, 4 villages will be selected randomly. Thus, there will be 16 villages from the selected Panchayat Samiti. Multi-stage sampling techniques will be used to draw the sample. At the first stage, 20 farm families from each of the 16 selected villages will be selected randomly into exploring the integrated farming system followed by the farm families. Thus, there will be 320 farm families from each selected family. One female member actively involved in farming will be considered as a sample of this study. Thus, the initial sample will be consist 320 farm women. Interview schedule techniques will be used for the data collection. And next is the result of my study. Table shows that maximum farm family but adopted crop and crop plus dairy with 41.2 percentage. Second identify farming system is crop plus dairy and vegetable with 28.74 percentage. Third farming system is crop plus dairy plus goatry with 12.4%, 12.14%. And fourth farming system identified is crop plus dairy plus vegetable plus food cultivation with 7.28 percentage. And fifth farming system identify crop plus dairy plus poultry plus poultry. Fourth farming system identify is crop plus dairy plus vegetable plus fruit cultivation with 7.28%. Fifth farming system identified is crop plus dairy plus goatry plus poultry with 3.23%. And six farming system were identified crop plus dairy plus vegetable plus floriculture with 2.83%. And next one, seven farming system identified crop plus dairy plus vegetable plus goatry with crop plus dairy plus vegetable plus goatry with 1.21%. Next farming system identified crop plus dairy plus fruit with 1.21%. Ninth farming system identified crop plus dairy plus vegetable plus goatry plus poultry is 0.80%. And next farming system identified crop plus vegetable plus fruit with 0.40%. And next one is crop plus vegetable plus poultry with the percentage of 0.40. And last, crop plus dairy plus floriculture identified with the percentage of 0.40. According to the farming system, the possible combination of the different farming system shows in the table where crop plus dairy is the one of the main component component of the crop plus dairy is one of the main component adopted by the farm families. These are the conclusion of the study. The study was purposely conducted in the Udaipur district of Rajasthan. The aim of the study to identify the existing integrated farming system adopted by the farm families. Such work is helpful for farmers to enhance their livelihood standard and income. It also helpful to for optimal utilization of resources through IFS. According to the research study, crop plus dairy and crop plus dairy plus vegetable was observed as the main profitable model in the Udaipur district. The extension agencies and NGOs needs to take care of other models, improve in farming techniques of remaining models, so other farm families also benefited. These are the references. Thank you, ma'am. Thank you, everyone. Thank you, everyone.
Thank you so much, Akanksha ma'am. Next, we have virtually connected with Mr. Roshan Raj. So next, on a topic of organic agriculture organic agriculture in India, and Roshan Raj Bujil. He's from Manipal Institute of Management, Manipal.
Thank you very much, ma'am. I have shared my presentation. Is it visible on the screen, please?
Yeah, it's visible.
Okay. Am I audible? Very clear? Huh?
Yeah, I can hear you.
Thank you very much. So, very warm welcome and very good afternoon to everyone present. Today, I am going to present on a review paper on organic agriculture in India. So, I am Roshan Raj Bujel, as ma'am introduced, a PhD research scholar from Manipal Institute of Management, Manipal Academy of Higher Education.
मुझे कुछ नहीं बस प्रेजेंट तो करना था.
There is some feedback noise coming from elsewhere. Yes, thank you. Okay, so to introduce, firstly, organic farming has always been present in India in the form of traditional farming. So this is not something new to India, but we had moved towards more production during the Green Revolution, where we use extensive use of fertilizers just to increase our production capacity. Though we did decently well during the Green Revolution, but we also saw many negative impacts of increasing production using synthetic fertilizers. And now there are various benefits of organic farming. So this paper systematically reviews existing literature on the methods, benefits, challenges, and the future of organic farming in India. And we also focused on vermicomposting and biofertilizers. So organic agriculture in India, what is the need for organic agriculture in India? Organic agriculture farming is a method, as you all know, of producing food without the harmful use of synthetic chemicals or pesticides or genetically modified organisms. So it is becoming increasingly popular, not just in India, but rest of the world as well. People are looking for sustainable farming methods, and organic farming is one of the most popular amongst all the sustainable practices. So firstly, what organic farming does is it improves the soil health and fertility, according to many literatures in this very topic. So it is also have been found to be an excellent method of pest and weed control and promoting biodiversity. So this is a very resilient and a sustainable method of farming, provided we are trained and we have the resources to practice this method effectively. Secondly, organic farming can be beneficial for farmers by reducing costs associated with synthetic inputs and increasing the value of their crops. As we all know that when we go to the market or any supermarket nowadays, the organic produce has a higher price than the conventional produce of the same variety, whether it be vegetables or food grains. So there is a premium people are willing to pay for organic produces in the market today because of the health benefit associated with organic food produces. So this is the very reason why we focused on this review. And it also helps to mitigate climate change, because climate change has become a very, very big topic and a very strong area of concern for everyone. So what the review suggested, the various techniques that are in use in India in organic farming, They are crop rotation, composting, intercropping, biological pest control, vermiculture, green manuring, integrated pest management, rainwater harvesting, and also farmers were given farm certifications also through certain agencies for their produces, which are a little bit difficult for a common farmer. But as producer organizations, some farming producer organizations have been certifying their products as organic in India. So now let's talk about the benefits. We just touched upon it in the introduction, but let's look at it as to what are the exact benefits of organic agriculture. As I already said before, it improves soil health. It increases yield. Initially, it takes about 5 to 6 years for you to convert your land from a conventional farm into an organic farm. And there is a process of, rejuvenating the soil. I will not go into the details, nitty-gritties of that. But once you have done that process, then after many papers have suggested that after a period of about 5 to 7 years, they have shown that there is an increased yield than when they were farming conventionally. And as I already mentioned earlier, this reduces the cost because now we are not using any synthetic material or synthetic pesticides, which all cost the farmers more, agriculture input cost. And instead, the farmer focuses on green products, which are available in his own farm through vermicomposting and biocomposting and green composting and other methods. So that way it reduces his cost and makes his farm more sustainable because he is self-sustained now rather than depending on other producers for giving him supply of fertilizers or pesticides. Okay, in India we use a lot of products, bioproducts that can be used as pesticides. Some are examples of that would be use of neem or of cow urine and Artemis vulgaris. These kind of things farmers have been using to fight against pests and diseases using organic agriculture. So of course the food would be much more nutritious and healthy. There are a lot of literature supporting this statement as well. And there is a positive impact on the environment because now we have reduced the use of chemical fertilizers, and that also affects the ecological balance of the farm, as well as it reduces global warming as well. And if done well, it could provide better livelihood for farmers. But this is a very thin line statement because we need to really work for that. And this is a challenge that we need to address as we move forward in organic farming in India. So then it promotes biodiversity, and also if we farm organically, then there is the impact on the climate change mitigation, as well as the sequestering carbon in the soil and reducing greenhouse gas effects from synthetic inputs, and it helps in mitigating climate change as well to an extent. So now, what is the status of organic farming in India? Okay, so organic farming in India, the most recent literature that suggested was the year 2019. I have figures for that, and India has about 4.8 million hectares of that is nearly 12 million acres of land under organic agriculture, and which is only about 2% of our total agricultural land. So we are still in the growing phase. We have a long way to go, and there is a lot of potential. But the government has been promoting organic farming in India through the National Program for Organic Production, which was launched in the year 2000. And this was basically done to promote production and export of organic products. So government is doing, certain, they have framed some policies, and they are working for, trying to boost the organic farming capacity in the country. Because there is need for that. That is the major reason behind that. And now the challenges that the farmers are facing in the organic agriculture in India. There is a lack of, access to markets and fair prices for organic products. Because many organic farmers in India are struggling to market their products. Even if they're organic, half of them are not certified, and they are not able to find a good market for their product, because a farmer cannot go to a market, which might be in a very far off place. He needs the market to come to him. So there needs to be access for him to, give his products to transporters or to carriers who can take his product from his farm to the markets where it would fetch him a fair price. But he needs the market to come to him, and that is not something very easily accessible to farmers at this moment of time. And there is limited availability of organic seeds and inputs, and the seeds and inputs are quite costly if a farmer wishes to buy as well. And biofertilizers are little bit expensive for a common farmer at this moment. He cannot afford it. So the only way forward for him would be to rely on green manure, cow dung manure, and vermicomposting. And there is difficulty in pest and disease management because the farmer is not very knowledgeable about various techniques of organically managing pest and diseases, and the farmer needs a lot of training in the ground. And there is a lack of awareness and education among the farmers about organic produces. Some are knowledgeable about it, but not all of them. And there is limited government support on the grassroots level. This is what the literature suggests. And there is certificates and standardization challenges, because certain certificates are very expensive for a common farmer, and a farmer lacks knowledge on how to certify the product organically. And also, organic agriculture has been found to be very labor intensive. So the labor input cost might increase for the farmer. So what are the initiatives taken by the government of India in this respect? Government of India launched the National Program for Organic Production in the year 2000, primarily to promote the production and export of organic products. It provides a technical and financial assistance to farmers for organic production, and also provides certification for the organic products, which is very important. You cannot just say that a product is organic. We also need it to be certified by a recognized certifying agency. And so the government is pushing and doing its bid, but more needs to be done in this respect. And also there is Pradhan Mantri Fasal Bima Yojana, which is an insurance scheme for the farm producers. And there is the Rashtriya Krishi Vikas Yojana, which was launched in 2007 to aim to promote agriculture and rural development and assistance. It provides assistance to farmer trying to adopt to modern farming techniques, including organic agriculture. And there is this mission organic chain value development for Northeastern region, because Northeastern region have been found to be very, 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 very viable for organic farming. So the government is focusing through various policies to boost organic farming in the northeastern states of India. Organic farming scheme for certification of organic products and the establishment of organic farming clusters are being done at a certain level, but still it needs to penetrate into the grassroots levels, and a lot of work needs to be done. There is a National Organic Farming Research Institute, which is basically established only to promote organic farming and to do a lot of research work on improving the farming techniques. And there is also the e-National Agriculture Market. Basically, this is an online marketplace which is made to market the agricultural products straight from the farmer to the market, to the wholesalers. And the national agencies involved in organic agriculture in India, the Agricultural and Processed Food Products Export Development Authority, the National Center of Organic Farming, even ICAR through the National Organic Farming Research Institute, Nofre in Sikkim, which is the first organic state which has been recognized by the United Nations. So in 2019, so we have an organic state in India itself, which is a model for the country to follow organic farming. So and also the ICR network projects on organic farming has been started a long time back. And there are also self-help agriculture groups which are promoting organic agriculture at the grassroots level. The future prospects of organic farming. So what are the future prospects of organic farming? It's very essential for us to, go into this, because only then people would be willing and be motivated to adopt organic agriculture. There is an increased demand for organic products. So this demand is only going to increase in the near future because of the awareness of sustainability, the awareness of health, and the recent focus on nutrition during the COVID time has stressed upon healthy produces. Healthy produces and organic produces are known to have a higher nutritional quality than conventional agricultural products. So the focus shifts to organic products, and there is a huge demand for organic products, and this demand is only going to increase in the near future because of the number of factors such as growing population, and not just growing population but growing urban population, and reducing farmers. The number of farmers have been reducing, and people migrating to cities have increased. And this will only result in the demand for products, and not just products, because now, since the income of people have also been increasing in the cities, and with our economy growing, there would be more need to fill this gap of supply and demand. And agriculture products would be top of the list because of the nutritional value as well as the health benefits associated with the organic products. And economic benefits, organic farming can provide benefits for farmers because now we can sell they can sell their produce for a premium. So already in the markets, we can see that the organic product are priced at a premium. So now there needs to be found a method to, share this premium value with the farmers, because at the moment the farmers aren't getting the value for their goods. So if the agencies work to boost the income of the farmer, definitely this would not just benefit economically the whole country, but as well as the farmers as well. Of course, there are environmental benefits to this, because now we are reducing the use of synthetic fertilizers and pesticides, and there have been ample researches on the harmful effects of fertilizers and pesticides when they are overused. Of course, we might have to reduce this, but it might be difficult looking at the demand for food. So if you look at food security, we need to still practice conventional methods until we have found an alternative that is better or good enough to meet the food needs and the food security of the country. And so organic farming is a way forward, definitely a way forward. It is a traditional agriculture that has been practiced by our ancestors from time immemorial, and it had been beneficial before until we shifted to conventional agriculture and our land became a little not so conducive for organic farming. So now what we need is to rejuvenate the soil, make the soil fertile again organically. And because sustainability for the near future is also a very important topic today, and environment is something we all have to think about. Climate change is very real. The monsoons in India have been changing. The climatic conditions are very adverse nowadays. So we need to find a sustainable way, and a traditional way, the organic way, is, I think, a better way. Even the literature support this statement. And there are also, health benefits of eating organic produces. They have higher nutritional quality, as I already said. Also, they do not harm this human system body as do the produces with the same with use of pesticides, because they have some certain side effects of using pesticides. And the government is taking initiatives on this, and we need to increase the exports of organic produces. And there is also need to develop organic clusters. There's a need for agroecological farming system and research and development. A lot of research and development needs to be done to increase the efficiency and profitability of organic farming in India. So overall, organic farming will help in more sustainable and equitable food system in India. So I encourage also the audience to support organic farming by choosing organic products and supporting farmers who practice organic agriculture. And finally, I would like to close by saying that agriculture is important aspect for sustainable future of India, and it can bring positive change in economy, environmental and health sectors. And we need to support sustainable farming in India. The population of farmers is reducing as the youth today in the rural areas are looking for alternative employment opportunities, leaving their villages and moving to cities. So we need to support the common farmer, and please, please, please respect the farmer who is, producing food for us. And because of them only, we are able to have a, sumptuous meal today. And I would lastly thank all the farmers of India. Thank all the farmers of India who are striving to, move towards sustainable practices. And with that, these are the references. And thank you very much. Thank you for the audience. Thank you very much.
Thank you so much, Roshan Raji. And it was really lovely. And yes, we have lots of respect for farmers. And we will have a lunch break right now. After that, Mushkan Dikshit. I guess Mushkan Dikshit is with us virtually. I request her to be present after the lunch. Give
Yeah,
us
sure,
30
ma'am.
minutes for lunch, then we will get back to you.
Okay,
Mushkan.
thank you so
Thank
much.
you.
Hello.
कैमरा को असला स्पार्क इस पे वेलकम बोर्ड ही है तो तुम्हारा नहीं कट देते नाम बोर्ड इधर ठीक है ना इधर बोर्ड डीएसएल का ठीक है बोर्ड तो है ना फोटो का असले देखिए
A very warm good afternoon. Am I audible? Ma'am, there's some issue. You're not audible. Excuse me, ma'am. Excuse me, ma'am. Am I audible? You are not audible to me. I guess the mic is mute.
Hello.
Hello.
Yes, ma'am. Can you hear me?
Yeah.
Okay.
Yeah. May I proceed?
Yes,
Actually, you were
sure,
not
ma'am.
previously audible, so I was not able to hear up. Should I start with?
Yes, ma'am. Sure. There were some technical issues. Right
Okay.
now it's
Okay.
solved. Please start.
No worries. Okay. Form that it is visible or not visible.
Hello. Will right
Hello.
now.
Yeah. Okay. Review on the study of disparate leaf diseases and analysis of methodologies used in leaf disease detection. This is Muskan. I'm from Chitkara University Institute of Engineering and Technology. I'm pursuing bachelor's in engineering from the domain computer science. This is our flow of presentation. We'll be starting up with the introduction, moving on towards the objectives, methodologies, discussion, and conclusion. Introduction. As we know that agriculture sector is the fundamental of all the sectors. Hence, it is important to protect the plant health. The physiological functioning of plants that are irritated by the prolonged pathogenic organisms, biotic or infectious disease agents is called plant disease. Pathogens are classified into fungi, bacteria, viruses, nematodes, powdery mildew, downy mildew, black spot, fusarium wilt, sooty mold, granule wilt, wildfire tobacco, fire blight, rusts, mosaic virus, tamping of disease are few of the examples of leaf diseases. To detect these diseases through human eye, there are lots of scope of error. So we need to discuss them out. Symptoms of leaf diseases in different plants. Powdery mildew, powdery white coating on the leaves is being observed on the plants grapes, apples, cucumber, peas, roses, flax, pumpkin, squash, watermelon. Sooty mold impends photosynthesis. It is observed in orange, lemon, fig, grapefruits, and gardenia plants. Downy mildew, the upper portion of the leaf discolor. The cauliflower, broccoli, grapevines, lettuce, salvia, pansy, cabbage, basils are few of the plants in which downy mildew is observed. Black spot, it is observed on roses. Occurrence of black spots is being observed. Fusarium wilt, wilted leaves, stunts plants, blackened stem roots. Beans, tomatoes, peas, danthus, sweet potato, banana legumes are few of the plants in which fusarium wilt is observed. Objectives. To study the role and application of prevailing intelligent methods like deep learning, machine learning algorithms in detecting leaf diseases. To conclude with a comparison of the most suitable method and their limitation. To discuss the challenges faced in detection of leaf diseases while using image processing. Before I jump on to the methodologies in image processing, I would just brief you up what is image processing. Image processing is more accurately defined as means of translation between the human visual system and digital imaging devices. It helps us to extract meaningful information. Moreover, it works on huge data set and provides information accordingly. Open libraries provide information and recognize the diseases much easier as compared to the human brain. What happens is, what is machine learning and deep learning? So artificial intelligence is a broad area of computer science that makes machines seem like they have human intelligence. Machine learning is a subset of artificial intelligence. It is a fragment of artificial intelligence which can perform the classification, recognition of patterns, which draw the inferences from the data and the predictions based upon the existing data. It improves the experience. Few of the examples of machine learning algorithms: CNN model, learning vector quantization, LVQ, stochastic gradient descent, SGD, naive Bayes decision tree, K nearest neighbor, KNN, support vector machine, SVM, and random forest, RF are few of the examples. Moving towards deep learning, it is a machine learning technique that is inspired by the way we are learning from examples. The set of algorithms are used to analyze the provided data and provides the output values within an acceptable range. Deep neural networks, DNNs, deep convolution neural networks, DCNN, transfer learning, neural network, autoencoders, restricted Boltzmann machine, RBM, are few of the examples of deep learning algorithms. What are the steps for image processing? Image acquisition. This is the very first step in which we need to capture the leaf using the camera or the digital camera, phone camera or the digital camera. These stored images are further used for the database using a specific path. Second step is image pre-processing. This improves the quality of image by removing unsought distortions. Third step is image segmentation. K-mean algorithm is one of the example. Image segmentation is a form in which dividing an image into different sub-images. K-mean segmentation is a method which is partition-based, one of the classical data mining algorithms. It clusters the objects closest to the another images by clustering. Feature extraction. Interesting part of an image from where the required informations are extracted is called feature extraction. Then we analyze the data and moving towards decision making. By comparing the data analyzed, we have a set of data, which is called data set. Then there is a decision making according to the observed details. We conclude the result. Synthesis of the methodologies. I have reviewed many of the papers in the following years, which have been done. CNN with efficient net architecture, a convenient model accessible through the telegram using efficient net bow, concluding the average accuracy of 78%. SVM classifier justifies that SVM is a efficient, versatile, and memory efficient SVM with DOH and DOG. Cotton plant was the plant on which they studied. Efficient self-learning in AI model. 80% accurate, better than SIFT, SURF, and FREE. These are the different technologies. CNN, the training accuracy of 72% and 12% testing accuracy provided by CNN with UI designed in Flask. FDA, an automatic recognition of the crop disease gave the accuracy of 90%. RGB imaging system based on AMG 8833 grind eye thermal camera sensor. This technique and estimates the pathogens per plant promptly with it. TBDR and DLDR using CNN. This concludes that TBDR is more efficient than DLDR as the sensitivity was of 0.98. DCNN using NAG algorithm, RCNN, YOLO, and SSD. The NAG algorithm model gives the accuracy of 97.62. YOLO V5 image segmentation technique. The evaluation is based upon the plant village data set, which achieves the precision of 99.7%. KNN, the application based on KNN gives rightness of 96.77, approximately 97%. CNN, SVM, KNN, decision tree, and naive Bayes. The CNN model using TensorFlow library gives a higher accuracy as compared to other methods. NestNet 34, NestNet 50, VGG 16, VGG 19, and DenseNet 121. The best output is given by the DenseNet 121 model of 98.31 with CAR. Now, here are some of the techniques which are being reviewed and inferences are being drawn by me. LXNet is considered to be the best model for deep learning technique. This disease stage detection assesses of disease and its application while exploring the transfer learning to improve the data size. Development scope of hybrid algorithms like genetic algo, optimization, and colony, etc. to improve recognition rate. Emphasis the importance of MLDL, the automated devices like UAVs and smart mobiles in the era of agriculture. DL framework provides HIS, hyperspectral imaging for improvised result. Automation helps pathogenists and farmers to prospect exploration. Moving on to the discussion, the impact of the leaf disease needs a consideration to study the betterment scope. To study the lead disease, deep learning, machine learning, image processing techniques plays a significant role. To transfer learning approach, pre-trained models are helpful in extraction. On the basis of above research, each method has its own rate of accuracy and each are significant. The CNN, KNN, and SVM algorithms are used widely and have higher accuracy on disordered samples. The major techniques for the detection of the plant disease are BPNN, SBM, K-means clustering, and SGDM. There are few of the limitations of these techniques used. While monitoring of the plant leaves disease in real world field conditions, we investigated background data effects on the resulting image. Detection of different infections for multiple diseases is difficult. Infection level is not determined through these techniques. Currently, available data sets are usually small and do not contain enough images, which is necessity for the higher quality decisions. Automation of the detection system using complex images captured in outdoors, lighting, and intense environmental condition is also very difficult. Conclusions. ML and DL techniques are used to analyze the healthy and diseased plant leaves and thus helps the farmers to diagnose the disease and to take appropriate actions on time. It is time efficient. High proficiency methodologies under DL frameworks and advanced imaging techniques of CNN are resolving the challenges of HIS hyperspectral imaging. The CNN convolutional neural networks, KNN, K-nearest neighbor, and SVM, sport vector machine algorithms are used widely and have higher accuracy on disordered samples. Thank you so much.
Thank you so much, Muskan ma'am.
Thank you, it was my pleasure.
May I request Harsh K. Shastri sir? Can you hear me? Harsh K. Shastri sir?
Hello, am I audible?
Yeah, Harsh sir. Harsh
Okay,
K.
madam.
Shastri.
Yes, madam. I'm going to share my screen. If it's visible or no, just inform me.
So can you hear me? It's me, Sonali, here to coordinate you. Okay, all right. So we have one more speaker, Mr. Harsh K. Shastri. His topic will be from Agriculture 4.0, or 4 revolutions of agriculture through plasma, the 4 states of matter. So he will be from Ahmedabad, Gujarat Technology University, Ahmedabad, India.
Okay, so good afternoon, everyone. Myself, Harsh K. Shastri, a research scholar from Gujarat Technological University, Ahmedabad. And today we are going to discuss about fourth revolution of agriculture, that is 0.0. And this revolution through the plasma, the fourth state of matter. We will see some of the recent development challenges and future scope. This research I'm doing under guidance of Dr. Manish Thakkar, that is head of the Instrumentation and Control Department of LD College of Engineering, with Dr. Suryakant B. Gupta. He is a research scientist at the Plasma Research Institute, Ahmedabad. So this is the outline of today's presentation. We will start with the objective. We will see what is agriculture, what is plasma, how plasma can elevate the agriculture or revolution in the agriculture, some of the recent research and major findings, challenges, future scope, and finally some conclusion. So a hungry stomach can't see what is right or what is wrong. Food is one of the most important parameters to survive, and nothing in the world can equate the piece of bread. By 2015, world population would be approximately 9.7 billion. That means we have to produce 70% more food that we are producing today. So definitely it's the right time to find a sustainable solution for the agriculture related issues, like to meet the future food demand while fighting the degraded or natural resources, climate change, food wastage, funding or less glamour in the field of agriculture, and degraded food quality, and the contamination is also one of the issue on which we have to work. So starting with agriculture 4.0. So in the 2015 United Nations World Global Summit was held, and it was discussion based on the to design a blueprint to achieve a better and more sustainable future for everyone. Outcome of this event is a 17 interconnected sustainable development goals, SDG. Basically, my research will targeting out of this 17 SDG, that is the goal number 02, zero hunger, goal 03, good health, well-being, goal 11, that is sustainable cities, and goal number 12, that is responsible consumption and protection. So to work on this goal, in 2018, World Government Summit was held, and in that summit, the term agriculture 4.0 was coined. Okay, so at that time, all the industrial people are working on the industry 4.0, but World Government Summit, all the government agree that we require a next revolution of agriculture. So what is agriculture 4.0, or what is the fourth revolution of agriculture? Before that, we have to see the past three revolution. So first revolution, that is blue revolution, where all the nations were working on the to boost the irrigation in the field of agriculture. Next revolution was a chemical revolution, where we are going to set up a very huge factories of fertilizer and pesticide. Third revolution, that is genetic modification and biological revolution. And now it's a time for fourth revolution, or agriculture 4.0. So agriculture 4.0 contains new approach for the production like urban agriculture and vertical farming, some innovative technique, desert agriculture, seawater farming, space farming, some contemporary concept like food sharing, crowd farming, blockchains, use of biotechnology, and apply advanced engineering solutions or apply advanced engineering techniques like drone, IoT, artificial intelligence, nanotechnology, nanosensor, etc. So agriculture 4.0 is nothing but it is incorporate the cross-industrial technologies and its application for the digital farming. So this is about some basic idea of agriculture 4.0. Looking at our topic, that is with help of plasma. How can we achieve, how can we contribute in this revolution? So in the school time, we have studied plasma is nothing but it's a fourth state of matter. If we provide energy to any solid substance, it change its state from solid to liquid. Some more energy, liquid to gas. And even if we continue providing energy, that gaseous substance converted into the plasma. These all we have studied in our physics or school time. Fine. So on the right hand side, we are having the classification of plasma. So based on its temperature, based on its behavior, based on its property, these are some of the classification of our type of plasma, that is thermal, non-thermal, ionized, magnetic, non-magnetic, etc. To generate a plasma, some of the techniques or instruments which we are going to use, that is barrier discharge, gliding arc, plasma jet, plasma torch, etc. So what happens in the plasma generation? While we are generating a plasma, the plasma discharge generate the reactive charge species like electrons, ions, and if we are going to use a various gas combination like oxygen and nitrogen, then RONS, that is reactive oxygen and nitrogen species, has been generated. These species works or doing some chemical reaction with our food or biological substance, and based on that, these are some of the area where we can use for the betterment of our society, for the betterment of agriculture. So plasma technology has been used in industrial, medical, nanotechnology, etc. But now it's a novel area to apply this technology for the seed treatment, for the plant growth, for the quality production, for the decontamination of the food, and to improve the shelf life or for preservation. Okay, so let's take some of the example. So first example, how we can use a plasma technology for the seed germination. Okay, so basically from this figure, whatever the plasma technology we are going to use for agriculture purpose, that is non-thermal plasma techniques. Okay, so wherever you are going to read the sentences like NTP, that is non-thermal plasma technology or non-thermal plasma processing. Okay, so for the seed germination, just look at the bottom of my presentation. So this is a diagram of a plasma reactor. So on the plasma reactor, we are having some electrodes here. We are going to give some high voltage and high frequency power source to that electrode. On the top side, we are feeding some gas. And to maintain a certain pressure or vacuum inside the chamber, we are having our vacuum tube. And on this plate, we have placed some seeds. Okay, so this plasma treatment generated plasma or treat these seeds, and what's its effect on the seed germination, that we will see later on. Okay, so these are the technology, that is NTP. Gas pressure we are going to use for seed germination, that is low or medium. These are some of the gases, and what is the result? So result is based on the treatment, positive effect seen in the seed germination and growth of the plant. Some of the examples. So right hand side, The radish sprout that is cultivated with oxygen plasma irradiation. So clear difference we can identify. Image I have captured from the references 6 and 70s. And also some of the effect or positive result found on the plasma on the seed germination. On the radish sprout, chili pepper, wheat, sunflower, soybean, pea, ajwain, basil, tomato. So while going through all the research, I can conclude that the growth and enhancement was found around 20 to 50% in these literatures. So these are some of the seeds and the plasma technology or plasma processing done on the seed. And right hand side, we are having some results. Okay. So some of the findings that how or what is the reason for this seed germination improvement. So plasma can generate a structural deformation on the subject on the surface of a seed. So from this structural deformation, the capacity for water absorption is increasing, and this capacity of water absorption boosting its germination. Okay, different gas combination can alter the germination time. For example, if we are using carbon tetrafluoride, it delays the germination. And if we provide another gas like cyclohexane, so germination time can be, just a minute. Okay, so germination time can be accelerate. Okay, apart from the seed germination, some other effect has been seen on the plant. For example, a treatment of a soybean seeds on the plasma treatment. So apart from the seed germination, it increases the protein and sugar level. Talking about a wheat plant, then haze tolerance has found increase. However, it's not true for all the plants, all the seeds. For example, if we are talking about mung bean, no significant or no noticeable growth has been observed. Some of the research or some of the literature is also claiming that this plasma treatment can possibly doing some can possibly doing in the seed. Some of the cases, the DNA damage in the pea seed has been observed. In some of the literature, the cell elongation of the protein was found in the first generation, but in the next generation, again, the protein cell found normal. Okay. However, all the study which I have gone through, these all are laboratory-based studies. So real-time field study has not been or very difficult to find some literature based on real-time field studies. Okay, so this was the first example, that is about the seed germination. Next example, talking about the safety and quality of food. So World Health Organization said that every year approximately 4,20,000 death occurs, and that is because of foodborne diseases. Now, who is responsible for the foodborne diseases? So that is the contamination of our food. Contamination can be classified into two ways. First is a biological contamination, bacteria, fungus, viruses, etc. There is a chemical contamination, that is the pesticides and fertilizer. So again, there are two methods. First is a traditional method. Second is some of the novel method to get rid of this contamination. Traditional method, like we are going to do some additive or treatment, and some of the novel method like ultra high pressure ionized radiation, gamma rays, X-rays, electron beams, etc. So one new term here, that is a PAW, that is plasma activated water. So this PAW technique can also work on the degradation, can also work on the decontamination of our food. So plasma activated water, what is it? So it is a product of NTP, that is a non-thermal plasma reaction with the water. In the previous case, we are doing some plasma treatment on the seed. Now in this case, we are doing a plasma treatment on the water, and that water is converted into plasma activated water. So this water is having a very rich diversity of highly reactive oxygen species and nitrogen. And these species are considered as a primary reactive chemical for the food decontamination. Okay, so NTP or non-thermal plasma is a chemical-free and eco-friendly method which kills the bacteria, bacterial spores, biofilms, virus, and fungus. The first successful study was conducted on the strawberry. The thing is that while doing a plasma treatment on the food, because of its surface or because of its texture, it's not possible to cover all the surfaces. So this problem can be solved using a plasma activated water. Okay, so as I have said, the main key parameter was RONS, that is reactives of oxygen and nitrogen species. So based on that quality of RONS, the effectiveness of a decontamination of a food can be directly correlated. So RONS quality is depends on which type of water we are going to use, what is our power, what is the position of electrode, which gas we are going to use, etc. So again, there are lots of options are available, and among them we have to choose the best combination for our dedicated food decontamination process. For example, uh, if we are going to use a normal air, so obviously it's a lower cost, but if you are going to use argon along with this normal air, then higher level of H2O2, that is hydrogen peroxide. So it is more killer compared to your normal air. Okay. Again, the activation timing and position of electrode, that is underwater, underwater, above water, or combination of both. So this everything is affecting on the RONS quality, and this RONS quality will define what level of decontamination could be done. Okay, another example of a plasma activated water, of other treatment. So here, the plasma activated water, and we are doing some mild heat treatment along with the plasma activated water treatment. So plasma plus temperature treatment. On the here, we are having a plasma plus ultrasound, bleaching plus thermostatically controlled. Okay, so while using this combinational method, some combination contamination found more effective in some of the cases. The references I have mentioned here. Okay. However, in some of the cases, several sensory deformities can be found on the food. That is the color changes or some texture parameter changes, etc. Okay. However, the majority of pesticide are going to degrade with this plasma activated water chemical reaction with the pesticides, and these are some of the pesticides I have found from this literature. Okay. So critical finding in research scope. So plasma activated water is considered as a less costly and more readily available. Thus, it can have some potential to use at commercial scale. As I have said that in the previous, whatever the finding was on the lab-based finding, so here there are chances that we can do some commercialization of this plasma activated water. Okay, DBD, that is another technology to generate the plasma, that is dielectric barrier discharge. So it is having a capacity to produce 520 liter per hour PAW, and this PAW can be stored. Okay, So no necessary that we are having some plasma generator. We can have this PAW from some research institute, and we can use for the decontamination of our food. So, however, the very low literature found on the development for the continuous process system. Optimization and precise mechanism to be identified is also a challenge, and majority of the time, the majority part of pesticide and other chemical pollutants are removed. However, 100% removal of the chemical pollutants is not, I have gone through any literature. Okay, so these are, we are talking about food safety and quality. Let's take one more example that is about how to improve the shelf life of the food. Okay, so again, World Government Summit report has said that 33% to 50% of the food produced globally is never eaten. Okay, so what happens to this food? So one, who is the responsible? So some of the bacteria, pathogenic bacteria, they are into some scraps. Okay, so two type of bacteria, that is gram positive and gram negative, based on the thickness of its wall. So gram negative bacteria is less resistant to the plasma treatment, for example salmonella, E. coli, etc. While gram positive bacteria, it is having a more resistant to cold plasma treatment. So to preserve the food, some of the traditional treatment like chemical treatment, we are washing the food with some chlorine, citric acid, etc. Thermal treatment, dehydration, or a coating treatment like in the apple, and we are doing some coating to preserve it for a long time. Okay. However, this treatment affects the appearance and textural damage of a food. Some of the non-traditional treatment like cold plasma treatment, dielectric heating treatment, and there are some other treatment I will mention here. Okay. So what happens in this treatment? So three parameters. First, this treatment either damages the cell membranes, second damages the intracellular proteins, and third damages chemicals of nucleic acid. Okay, so the plasma radicals, especially hydroxyl radicals, is having some toxic effect, on the proteins, on the lipids and the proteins, and the protein damage. And this damaged protein damages the cell spore and damages the nucleic acid. Okay, so another example to improve the shelf life with help of a plasma. So Contino has worked on the milk pasteurization and milk treatment on the or plasma treatment on the milk. So some of the result, NPG, nanosecond pulse plasma generator, and target is a bacteria. So bacterial count is degraded by Firlock cycle. Talking about E. coli, then dielectric barrier discharge plasma mechanism, and reduces the E. coli salmonella count by 2.4 log cycle. The references I have mentioned here. So finally, we are having some, I have some image collection. So left side, the for plasma activated water on the xenia. On the middle and the right hand side, we are having some clear comparison with the plasma treatment of sweet basil, that is our tulsi, and the research has been research is going in the New Jersey Seton Hall University. So on the left side, we can see the control basil, that average height is a 400 sorry, it should be a 440 millimeters, and the right hand side, the average height is compared to lower to the treatment one. Same way on this side, we can see the plant growth. So plasma treated and non-treated plant. Okay. So let's conclude. So to meet the future demand, the fourth revolution of agriculture is essential. Cold plasma technology and plasma activated water have potential to boost the seed germination, seedling growth, and shelf life improvement. Cold plasma processing can significantly enhance the essential oil and antioxidant level on the herbal medicinal plant like basil. Okay, so this reference is the same reference that Seton Hall University. Okay, so in this research also they have claimed that the antioxidant level of of a sweet basil has been increased in certain scales. Okay. However, the challenges of 100% decontamination food and the technology is novel and looking at the food diversity, the literature finding is just a tip of an iceberg. Most of the research are experimental types and whatever the result we are getting based on some experiment. However, it's very difficult to find any simulator or simulating kind of studies for this technology. And whatever the research or product development, if we are going to measure on the TRL scale, that is a scale developed by the NASA, that is the technology readiness level. So most of the research found up to 4 to 6. So it's also having a huge scope to bring the current finding to TRL line, where final product can be launched for the operational environment. These are some of the references I have used in my presentation. Thank you.
Thank you so much, sir. Harsh, we are talking with him from Gujarat. He is from Gujarat Technology University, Ahmedabad, and yes, it was really lovely to know about the plasma treatment and how the plasma support the agriculture. Thank you so much, sir.
Okay.
And next, I would like to request Pankajini Shamal. Her topic will be molecular dissection of Sith B light tolerance in rice. She is from ICAR NRRI Katak. Over to you, ma'am.
Hello. Hello.
So over to Pankajini Shamal.
Hello. Sir, is it okay? Good afternoon, everyone. I am Dr. Pankajini Samal. Now I am working as a senior research fellow at ICR National Rice Research Institute, Cuttack. And my today's topic is molecular dissection of sheath light tolerance in rice. Before going to the whole presentation, I will briefly describe some of the outlines of my today's presentation. I have divided my presentation into 4 parts. Actually, here I will be delivering on one most devastating disease of rice, that is called rice sheath fly disease. In part 1, I will be focusing on the screening of rice genotypes against sheath fly disease, and then for mechanism, and then for resistant mechanism, I will be delivering on transcriptome analysis, QTL analysis, and proteomics analysis. Coming to the introduction, actually rice doesn't need any introduction. As we all know, rice is the staple food for more than half of the world's population. And we also know population is increasing day by day, so the food demand is more. We have to produce 40% more rice in order to meet the rice demand. And also, most of the high-yielding rice varieties are susceptible to various abiotic and biotic stresses. In abiotic stresses, that may be drought, salinity, cold, or heat stresses. But my focus is on the biotic stresses. That is one fungus called Rhizoctonia solani, which causes up to 50% of yield loss to rice cultivation. Why sheath blight? Why I have chosen this disease? Because till date, there is no major resistant gene or any resistant source has been identified to combat that disease. Very few reports are available on host pathogen interaction. For formulating proper management strategy, we have to focus on more on how the disease can be properly managed, and we need more focus on understanding the mechanism of its resistance as well as the mechanism on its susceptibility. If the one rice genotype is susceptible, then why it is susceptible? We should know the genes behind the susceptibility function or resistivity function. We should act from both the sides. That's why we have to understand the disease etiology, why this disease occurs, and The causal organism for sheath blight disease is Rhizotonia solani, and it is a polyphagous fungus. It can rely on any kind of host. That's why it is very devastating in nature. And for the favorable conditions for disease development is where high relative humidity, high temperature, and where high levels of nitrogen fertilizer were used. And when the crop canopy are very dense, then it will altogether provide a favorable condition to increase the disease. And also the fungus can increase its number in close plant spacing where more than 2 seedling per hills are present. Plants are more vulnerable to sheath blight during the rainy season and at active tillering stage. Through sclerosia or infected bodies in floating water, it can easily transfer from one land to another, and it causes the infection. Actually, rice is we basically depend on irrigated water for rice cultivation. Through irrigation of water channel, that sclerosia move from one field to another, causing the infection, spreading the infection. The symptom is commonly known as snake skin disease. In Odia, all call them Sapakati rogo, as the symptoms resembles with the snake skin of Rossus viper. These are the symptoms you can see in the screen. This is the symptom lesion on stem. From the rice stem, it starts spreading towards the tip of the towards the panicle, and then it attacks the panicle, causing grain yield loss and poor filling of grains. Then coming to screening of genotypes, actually we have collected 200 rice genotypes, and we have screened against R. solani for identification of resistant source, and we include some of the resistant rice lines of international check like Tetep, Jasmine 85 and taking, and some of the susceptible varieties like Sonnasavan, Tapashini, Annapurna and Lemon. Then we have done the screening on the basis of a protocol provided by ERI that is called relative lesion height. The relative lesion height is the average vertical height of the uppermost lesion on leaf or sheath expressed as a percentage of the average plant height. This is the scoring scale of rice sheath blight disease. 0 means there is no infection. 1 means immune or resistant, and as usual it is 3, 5 and 7, and 9 is the most susceptible one. Then actually we have artificially inoculated the rice plants with R. solani, one of our own culture, that is CRRI RS8. This is the whole methodology of artificial inoculation. We grow the inoculum in a plate, then we transfer it into the transfer for multiplication in rice straw beads. Then the inoculated straw beads were tied on the leaf sheath area of rice plant. And then after 8 to 9 days, we can observe the disease is spreading from bottom towards the top. From the screening data, we have identified CR1014 and Tetep with disease score 3, and so we call them, these are the two tolerant varieties. And Swarnasavan, Annapurna, Puja, Tapashini, Navin, Lemon, Jasmine 85 and taking showed susceptibility reaction with a disease score of 7. These are the representative photographs of 10 selected rice lines, and this is just the graphical representation of 10 rice genotypes infected with R. solani. From the earlier data, we have selected 2 indica cultivars, CR1014, one of the CRRI's own rice variety, and Swarnasavan as the best tolerant and susceptible variety for studying the QTL analysis. Actually, today's my topic is basing on starting from genomics, then transcriptomics towards proteomics. Then coming to first part, that is called genomics, we did the QTL analysis for sheath blight tolerance in rice. For QTL analysis, we have taken CR1014 as the tolerant one and Swarnasavan as the susceptible one. Again, we screen the disease progress curve you can see here. In case of Sonasoban, the disease progress is quite higher, and in case of CR101 for the disease, after 14 to 15 days, the disease shows the disease not progress well as compared to that of Sonasoban. We have also confirmed the bioassay with a detached leaf assay. Here, we perform the detached leaf bioassay by taking 0 hour, 24-hour, and 48-hour and 72-hour post-inoculation method. Here, the mycelia growth appeared within 24 hours of incubation, and lesions were clearly visible within 48 to 72 hours. You can see here, in case of Sonasoban, the lesions are clearly visible after 48 hours, showing susceptibility reaction, whereas in case of CR1014, up to 72 hours, it shows clear-cut resistant reaction to Arsolani as compared to that of Sonasoban. On the basis of the above results screening data, we have developed two mapping population. One is Sonasoban CR1014 and one is Taposhini CR1014. And these are the generation we have maintained till now. From this, Sonasoban CR1014 population was used for QTL analysis. These are the frequency distribution curve of sheath blight disease response in terms of RLH of different mapping population in different season. From the above study, the significant finding is mapping of one minor QTL, that is QSHP 1.1 of chromosome 1 with link marker RM11935 and RM11968 covering 0.51 MB region, which was the QTL region, and we thought that this region might be the reason behind the resistivity of CR1014 as compared to that of Sonasoban, and that work has been published in Euphratica. And then coming to the fine mapping of that QTL region, QSHP 1.1. In QSHP 1.1 for fine mapping, 18 SSR marker from the Gramini database and 138 indel markers were designed using RICE SNP-seq database. And out of them, 13 markers were found to be polymorphic and used for, again, QTL analysis to know the particular region of resistance in CR1014. Here, we narrow down the QTL region to another marker we got, that is RM11945 with the reduction in QTL region. Then now the region is 0.48 MB, and in 0.48 MB we have to predict the candidate gene. This is gene prediction in the QTL region. We have predicted 81 genes in that region, out of which 11 genes were found to be disease responsive. Among 11 genes, through RT-PCR, QRT-PCR, we confirmed 3 genes, ubiquitin domain-like phosphatase protein, receptor-like protein kinase, and powdery mildew-resistant protein. These 3 were found to be the candidate gene, and these 3 genes will be further analyzed for future perspective. Then coming to the validation of QSHP 1.1, also the QTL QSHP 1.1 was validated in the backcross population, where the presence of QTL shows resistant band in 8 resistant lines, and the absence of QTL region shows the susceptible band in the 8 susceptible lines. That is one validation our QTL QSHP 1.1. This is the publication. Then the genomics part is over. Now coming to the transcriptomics part. In transcriptomics, these 2 lines, CR1014 and Sonasoban, were used for transcriptome analysis to study, and the leaf samples were collected at 3 days and 9 days of inoculation to study the early and late expressed genes. The transcriptome profile of both the genotypes were compared through high throughput RNA-seq on Illumina HiSeq. This is the basic step used for transcriptome analysis. there is a summary of the red alignment where we got more than 90%. These are the upregulated and downregulated gene at various time points after inoculation with R. solani. This is just the Venn diagram, just the representative Venn diagram and epichem plots of differentially expressed genes. And these are the differential expressed gene between some control and treated line of CR1014 and control and treated line of SONOS01. Also, we have done the functional enrichment analysis where all the raw data generated from transcriptome analysis were gone through functional enrichment analysis. That is called gene ontology analysis. In gene ontology analysis was performed to classify the sheath-light responsive DGs into three functional categories, that is biological process, molecular function, and cellular component. Most of the DGs from both the cultivars were involved in biological processes such as response to stimuli, immune process, signaling, multi-organism process. Also, we have done the gene regulatory network analysis on the basis of cytoscape, where the predicted gene-to-gene interaction would help us to better understand how the coordinate expression of multiple genes play important role in regulating sheath-light disease response in both the cultivars. In our investigation, the GNN analysis shows the co-regulation of DGs like cytochrome P450, senescence associated gene, glutathione specific gene, flamin monooxygenase, receptor kinase, and disease-resistant protein. These can play an important role to combat the deadly sheath-light disease. And the transcriptome data has already been submitted to NCBI in the following ID. Then after the shortlisting of the DEGs, coming to the validation of differential expressed genes through qPCR. Many DEGs were observed and among them 63 genes were selected for QRT-PCR validation. For 63 genes, 20 genes showed resemblance expression with the RNA-seq data. This is just the RNA photograph in agar gel. This is the validation data, validation of differential expressed gene through QRT-PCR. Here the 20 genes showed resemblance expression with the RNA-seq data. From these 20 genes, any one gene can be the candidate gene for sheath-light tolerance. This is just the graphical representation of these 20 genes. From the 20 genes, 2 genes, terpene synthase and NB cellular disease resistance protein, showed some indication regarding the tolerance mechanism against sheath-light in CR1014. Here you can see in case of NBA cellular, in case of susceptible line, it is completely the expression is 0.
Hello, check.
Interestingly, the results showed the presence of the gene only in CR1014 and in tolerant nil lines. That means that gene is carry forwarding to the advanced generation. This data has been published in Protoplasma. Again, we have also done the proteome profiling of these two genotypes. Here we can see these are the differential express proteins, and we have just shortlisted some of the important DEPs like peroxidase precursor, thaumatin, NADP dependent oxidoreductase, glycosyl hydrolase. Actually, the highlighted ones are in resemblance with the transcriptome data, and these genes will be analyzed further. Also, we did the defense enzyme study. Here, the activity study of the defense enzyme like catalase, peroxidase, polyphenol oxidase, superoxide dismutase in both susceptible and tolerant plants were studied and analyzed. All these results showed the
Check.
differential defense response of tolerant and susceptible rice genotypes infected with R. solani at mRNA and protein level, also in genomics level. These findings have significant implications for understanding the rice and R. solani interaction and developing strategy to better manage the deadly disease. From this study, we for future we can propose the cloning and functional characterization of candidate genes or proteins from this study, and also we can develop development and validation of gene-based markers for the candidate gene that can be used in resistant breeding purpose for developing a high-yielding resistant variety for Shithla disease. These are the list of my publications. These are the abstracts and some extended summaries I have attended through conferences. These are my references, and these are the backbone of my studies. They are the backbone of my studies. Here are my mentor, Dr. Arup Kumar Mukherjee, Dr. Meera Madam, Dr. Kutub sir, and these are my lovely and supportive lab mates. Thank you.
Check. Thank you so much, ma'am. Do you have Rahul Adhikari here? Rahul Adhikari and Priyadarshini Sangamitra. Rahul Adhikari and Sangamitra. Okay. Ten minutes of tea break.
Hello, hello, hello. Mic testing 1, 2, 3. Come on, Dana. अरे hello, hello, hello.